Hello, everybody. My name is Jiwon Kim from Seoul National University. And on behalf of the Korean Chemical Society, I'd like to welcome you to the afternoon session called ACS Publication Summit at 2023 KCS Spring Meeting. And this session is hosted by American Chemical Society and Korean Chemical Society in the hope of bringing two societies together in the time of post-pandemic. And we have invited prominent scientists, especially in nanoscience and nanomaterials, and they are both highly respected scientists, and also they are the editors of American Chemical Societies, which includes Nanoletters and ACS Nano, and advisory boards of ACS Central Science. And this session is made out of several sessions, and I'll be chairing the first session, which includes the opening remark from the president of our Korean Chemical Society.
Okay, thank you. My name is Sung-min Shin, president of the Korean Chemical Society. On behalf of the Korean Chemical Society, it is my great honor and pleasure to welcome all my fellow chemists and participants to the ACS Publication Summit at the 2023 Korean Chemical Society Spring Meeting, entitled Post-Pandemic Transformations and Challenges in Nanoscale and Materials Chemistry. As we emerge from the challenges of the pandemic, we find ourselves at a pivotal moment in the development of scientific innovations for a sustainable future. The transformations and challenges facing us are significant, and it is essential that we come together as a community to exchange ideas, share insights, and collaborate on the frontiers of chemistry. At this summit, you will have the chance to hear from leading researchers from Korea and beyond, including editors from Nanoletters, ACS Nano, ACS Central Science, Council Chemical Research, and JEX-AU. You will also have the opportunity to network with your peers, meet with outstanding researchers, and discuss your work with others who share your passion for science. This summit promises to be an exciting opportunity to explore the latest groundbreaking research in this rapidly evolving field of scientific exploration. I also believe that through gatherings like this, we can strengthen our bonds and develop meaningful relationships to overcome challenges together. This event represents a significant step forward for future cooperations and collaborations between ACS and KCS. I hope that everybody enjoys the stimulating talks and discussions during the summit. Finally, I would like to express my appreciation to Professor Sung-ju Hwang and Jamin Nam for organizing this wonderful event. Thank you very much.
Okay, thank you very much. The opening remark will be given by Professor Teddy Odom from Northwestern University. She is currently editor-in-chief of Nanoletters. Please.
Thank you for joining us for this summit. I'm very excited for the program that has been put together. And I also want to thank KCS President Suk-min Shin for welcoming us and allowing us to be part of this important meeting for chemistry in Korea. That's, we don't take these invitations and this organization lightly, and we appreciate your allowing us to be part of it. Before we get into the speakers, I just wanted to say a few words on what I think makes ACS publications so special. Part of it is a big focus of ours is that we want to support authors. And so we as editors see our role as providing the best service to shepherd and promote your work. We're author focused. This is what we aim to do. And also what ACS publication tries to do is to provide training for both authors and reviewers, and to be leaders for the scientific community. And there are many educational opportunities, for example, to learn how to be a good reviewer, how to write papers, how to tell compelling stories. And so if you're not aware of these efforts to build up the scientific community, I highly encourage you to take advantage of these resources. We put a lot of effort into them, and I even have my own students watch these videos so they can prepare better manuscripts. Another thing that I think makes ACS publication so special is that the editors are practicing scientists, meaning we understand what it means when a paper is accepted, when a paper is rejected, when it takes a long time for it to be published. We get it. So when we're not doing well, you can complain to us, that's okay, but we'll do our best. You know, we're trying to do this. And I think this is also what makes ACS publications and the editors so special. And so as you'll see today, we have an international set of editors that tries to represent both the scope of the chemistry publications that we have, as well as the international reach of who we want to serve. We try to make that as representative as possible, and you'll see that, and I hope you'll appreciate that in some of the talks today. So I don't want to take up any more of the speaker's time, but thank you again for welcoming us to be part of this session. And we look forward to a terrific meeting of really this global excellence in chemistry and nanoscience. Thank you.
Thank you very much.
Good afternoon. My name is Sojung Park. I'm at Ewha Women's University, and I'm serving as an associate editor for ACS AMI. I'm very glad to be a part of it and chair the first session today. Today's first session, Professor Taek Han-hyun will open the first session today. He's a professor at Seoul National University and the director of the Institute of Basic Science. And until very recently, he has served as an associate editor for JAX for 11 years. And he's now on a advisory board for ACS Central Science. Please welcome Professor Taek Han-hyun for the first speech.
All right, it's good afternoon. It's my great honor to give some of my stories for the last the 5 years we have been doing in our IBS Center for Nanoparticle Research. Okay, I think it's on. All right, great. Let's get started. Okay. All right, so basically for the last the 10 years, especially as in our IBS Center for Nanoparticle Research, we've been working on designing new nanostructural materials based on the uniform size nanoparticle and their assembly, and eventually we apply them for the medicine area and also energy and catalysis. This topic, today's topic gonna be focused on that, energy and catalytic applications, especially in collaboration with Professor Young-Won Sung, who is currently serving as a deputy director of our IBS Center. And also last part will be a collaboration with Professor Tae-Young Kim, another a deputy director of our IBS Center. All right, so let's first start with fuel cell electrocatalysis. As you might know, fuel cell is along with the batteries, right? This is for the really important for the vehicles, right? Cars. So and using the hydrogen, fuel of 21st century. The only side product is the water, as you might know. But there's a big issue is that we have to use a platinum-based, the materials, right? Which is really expensive and rare, right? So and as you can show here, right? The more than half of the the fuel cell stack is coming from the cost is coming from the catalyst. As I just mentioned, that's because of platinum-based materials, right? So as you produce more and more, still I think platinum is there. That's why, I mean, it's a really big issue. So one of the issues, I'm gonna give two examples here. First is like, I mean, we're gonna use the less platinum by making alloys, especially like, I mean, the transition metal alloys. With the first example is the alloy with the iron. So we first made an iron PT alloy nanoparticle. Basically, we use half of the platinum that is shown here, right? So, but the problem is that a synthesized iron PT alloy nanoparticle is useless, catalytically inactive, disordered FCC phase. So to make it catalytically active, you have to heat it up at high temperature. But the problem is that, as shown here, when you heat it up high temperature, severe agglomeration of nanoparticle occurs, which is losing surface area, eventually losing electrocatalytic activity, right? So we made some trick here, which is actually before the thermal treatment. We actually coated polydopamine on top of the IMPTL nanoparticle and it performed heat treatment. As shown here, even after, I don't know how this is gonna work. Okay, so not working. So, this is better one. I have a bigger arrow here. So as shown here, is after like heat treatment, severe agglomeration. But after coating with the polydopamine, this polydopamine is during heat treatment transformed to the end of the carbon shell, which prevent the agglomeration. That's why particle size before and after is virtually the same. That's why we could get very high electrocatalytic activity as shown here. This is half cell data. More important is the full cell. We made MEA, membrane electroassembly, full fuel cell. And as shown here, amazingly enough, very high activity, allowing the very good stability as shown here. Even after 100 hours of continuous operation, only less than 3% degradation of the catalytic performance, which is really amazing. So that's why, I mean, after we published paper in JAX, several years ago, a lot of automobile company like Toyota and the Motors showed great interest in our result. We keep on working on this. And one of the issues is the synthesis of IMPT alloy nanoparticle. Actually, we adopted from the science paper 2000 by the Chris Murray. Now he's in the UPenn, as you might know. So the problem is that we cannot make stoichiometric 1,2 alloy nanoparticle in large quantity. So we come up with one of our Indian postdoc, Arun, came up with a very simple idea using simple inorganic chemistry. So we actually pick up the 1-ton alloy mixed metal complex of the iron PT as shown here. That is a precursor. And we just put it on the carbon support and just simply heating up. And then as shown here, we could in situ generate metal PT alloy nanoparticle as shown here, around like less than 5 nanometer in particle size. And as you can see, again, great performance here. Electrocatalytic performance here. That's what it reported several years ago, 2020 JAX. And then, obviously this can not just the iron PT, and we can also extend to the nickel PT, cobalt PT, and that kind of stuff. So here, especially early this year, what it reported in the ES is that actually now we could make a very special cobalt PT alloy nanoparticle using our 2020 JAX method, direct synthesis of a cobalt PT alloy nanoparticle, all right? And we could get the very high catalytic activity. Actually, just going to go to straight. MEA performance is shown here. We beat the 2025 DOE target here. Not all the parameter here, but in terms of mass activity, we actually for the first time beat the DOE target as shown here. That's what it reported in the ES early this year. So we keep on working on this project. In collaboration with Professor Young-won Song, and we hope we can actually commercialize this material for the real fuel cell vehicle applications. That's what we are aiming for. Okay, let's switch gear a little bit more fundamental materials chemistry or science here, which is actually, as you might know, a lot in the catalysis and a lot of energy devices, interface control is really critical. defect control is quite important, right? But oftentimes it's not easy to control defect, right? So and we published several papers in the long time ago in Science, and I just want to focus on the rather recent like Nature papers in collaboration with my friend, Paul Alivisaros. We collaborated like 7 years for this project. And as you might know, Paul was he launched like Nano Letters as served as a long time as editor-in-chief. Anyway, so here what he tried to do was we tried to make a core shell nanocrystals, heterostructure core shell nanocrystals here. Especially what we picked up was cobalt oxide nanocubes, and we decorated with manganese oxide truncate octahedron-shaped manganese oxide shell here. So here, manipulating chemistry here, all right? Doing, the maneuvering around that chemistry here, we can actually control the number of the shells here. There's 6 phases in the cube, right? So we can decorate only one phase. We can decorate 3 phases, and we can decorate all 6 phases too. And also we can control the size of the shell, size of the manganese oxide shell here. So by doing this, we can actually control the size and number of interfaces here, number of defects. And what we found is that oftentimes, people are interested in the hetero interface, right? What I mean by that is right here, right? In between cobalt oxide and manganese oxide shell, people are often interested in the hetero interface. But we found that yes, hetero interface is important, but we found is that there's another very important interface and defect is generated right here. In between manganese oxide shell, right here, there's a new defect called as a discrimination was generated here. So as I mentioned earlier, because we can control number of manganese oxide shell and also like a thickness, size of that manganese oxide shell, we can actually control number and size of this discrimination defect. So and then we can actually quantify that by using the computer vision technology analysis of this TM high resolution TM images. And then this is we are about to submit this paper actually. So we could correlate this with again fuel cell oxygen reduction reaction catalyst, electrocatalyst. That's the catalysis. That's what we've done here. So in continuation of this work, okay. So here we try to use a non-platinum oxide, mixed metal oxide as a fuel cell oxygen reduction catalysis here. So here, what we have done is again, this is continuation of the paper, our Nature paper 2020. So basically here, pick up the cobalt oxide cube, the same thing as a Nature paper, and then we decorate with a graded structure. So to minimize strain between cobalt oxide core and the manganese and cobalt oxide mixed metal ferrite, shell structures, oxide shell structures. We actually use the continuous a gradient shell structure, which actually use that for the quantum dot television produced by Samsung Electronics. They use a gradient shell structures, right? Similar thing is here. We got an idea from there. So as you can see, composition is slowly changing from the core to the the right outside the surface to the of core to the to the surface. So by doing this, we can minimize strain exerted in between this hetero interface, all right? So and then we did a bunch of different kind of characterization, using the X-ray of substance spectroscopy, TEM, blah blah blah, and then we could actually find that the descriptor, the deciding factor for this ORR activity turned out to be like a manganese, electron transfer from manganese to cobalt in the octahedral site. And then we turned out to be that we could optimize the best activity is half and half. Cobalt, manganese, half and half. Oxide gave the best performance. As shown here, even compared to standard PT catalyst, our system exhibits a very good stability That's because we minimize using the gradient like a shell structures. That's reported like in nano letters. Yeah, nano letter, Terry, nano letter, yeah, that's right. And then it's a continue. We also did depth calculation justified our explanation. And then in continuation of this research using the again ORR. So basically here is that we actually designed multimetallic spinel oxide structures here, especially like a ferrite here. So as you might know, spinel is the AB2O4 structure, right? Here is a Fe2 is fixed, and the A site is we actually use the mixed matter. You could vary. So that as you might know, this is our most important paper. Around like a 5,000 citation, something like that. So using this our heater process, we could actually fine control the composition of the like A site here. So here is a mixed metal cobalt and manganese. We can continuously vary from pure cobalt ferrite to the pure manganese ferrite. We can control it. And then we can fix the size. We can get the same size, but simply changing the A composition. And then we could study where is the maximum activity, where's the best activity, and turn out to be like 1, 2, 3, 25 and cobalt 25, and and we also like perform different kind of XF analysis along with like ion mass power, and then we could actually study what is the descriptor here. Turn out to be again manganese octahedral site is the major contributor for this ORR reactions here, and turn out that the best activity is cobalt 25 and manganese 75 gave the best activity. All right. Okay, let's switch gear a little. The ORR reactions I just mentioned in the fuel cell ORR is a 4 electron reduction process. So O2 plus 4 electron plus H2 gives H2O. That's the the fuel cell electrocatalysis, right? But when you stop in between, just 2 electron process, O2 plus H2 gives H2O2, which is a very, very important chemicals and a lot of different kind of applications. More recently, especially this is a cleansing solution for semiconductor fabrication process. That's why this H2O2 became really, really important. But the way they make the H2O2 is as shown is really terrible. So-called anthraquinone process as shown here. They use a palladium high pressure, high temperature reaction, which is really totally expensive and environmentally unfriendly process. So one of the issues as shown in our review article and another ACS energy letter here. So the people try to use the electrochemistry here. So 2 electron ORR reactions to get H2O2. So here, what we have done is we try to learn from the nature, which is when you look at the H2O2 is produced in our body using the so-called superoxide dismutase, and which is actually that looks like this. SOD is a representative antioxidant. You can get it from not from Walmart, but anyway, I think you can get it somewhere else. Okay, so basically here is that this is porphyrin, metalloporphyrin structure, right? Try to mimic metalloporphyrin structures. We pick up graphene oxide, and then we actually dope the nitrogen first, and then putting different kind of different kind of metals. And the Jongsuk Yu as Universal Seoul did the depth calculation, turned out to be the best metals are cobalt. So we put in cobalt. Turned out to be cobalt is really unique chemistry here. And then we could make something like the metalloporphyrin like structures using graphene oxide doped with 4 nitrogen here, I just shown here. And then with a bunch of different calculations, turn out to be single cobalt atom is dispersed on this graphene oxide like a matrix here. That's what we could make. And then as shown here, tremendously high activity is shown here. Even compared with a typical the PGM based electrocatalyst, gold or silver or platinum based catalyst, our catalyst is order of magnitude higher H2O2 electrocatalytic activity is shown here. And then also this is highly stable. Even after over 100 hours operation, there is a minimum activity loss as shown here. And then we continue work on this project, and this year, early this year, we reported Nature Catalysis with Ted Sargent in University of Toronto. I think it says Ted becomes your colleague now in Northwestern, right? So yeah, basically here is that we changed the material slightly, which is actually we pick up the like a carbon nanotube, and then the key thing is the oxygen treatment here. So oxygen, so the incorporating something like the imparting some kind of oxygen, moiety, oxide moiety on the surface of carbon nanotube, we could get like a very good adhesion of this metallophthalocyan on top of this oxygenated carbon nanotubes. Okay, again, we did depth calculations and optimize. Again, here, turn out the best matter is cobalt again. All right, there's something unique about the cobalt, and then and turn out to be this gives now metal cobalt phthalocyan is sitting on the absorbed on this oxygenated carbon nanotube structures, and as you can see, single cobalt here, and give a really high activity here, as is shown here. Compared to the pure carbon nanotube, a lot better, 10 times higher activity. And also one of the issues can you make a concentrate H2O2 with high selectivity? So competing is a 4-electron process to make a water, right? So you try to make a more concentrate, selective formation of hydrogen. That's what we have achieved in this, the Nature Catalysis just published just 1 month ago. All right. So we can actually we can continue work on this process, and hopefully, as shown here, we could get the concentration of 5 or 8%, which is very high, very high. So actually the one unit for the semiconductor fabrication cleansing solution is around the 7 to 8%. So we are approaching to that right now. So that's what you have reported here. Okay, let's switch gear and talk about the It's like I've used a. Oh, good, plenty of time. All right, good. So in the final topic, I'm going to talk about photocatalysis. All right. So the first topic I'm going to touch on is the paper we published several years ago in Nature Materials in collaboration with Kit Nam at Material Science of Seoul National University, and also Hyungjung Kim at KCS Chemistry here. So basically here is that as you might know, as I just mentioned, a fuel cell, PMFC, right? Fuel cell is using the hydrogen as fuel. As you might know, hydrogen is the fuel of 21st century. Why? That it only produced as water as a side product, right? The most clean. But the way you make hydrogen is not clean at all. It's a totally disaster in terms of it's totally environment unfriendly process, right? Which is actually that the cooking natural gas, which is major component natural gas, as you might know, methane, right? At high temperature, react with water at high pressure and high temperature, right? And produce a bunch of CO2, right? And also this is this is how you make hydrogen, right? So the people try to make the hydrogen a different way of doing it. One ideal way of doing is what? Photocatalytic water splitting. There's a lot of work has been research in the chemistry in general. There's so many publications on this water splitting, photocatalytic water splitting to generate hydrogen, right? So again, we try to learn from the nature, right? Which is enzymes, right? As shown here, hydrogen enzymes, bacteria, can produce at room temperature, 1 atmosphere, right? Hydrogen, because of this enzymatic cooperating interactions, right? So what you try to do is, can you make something like enzymes, right? But one of the biggest problem enzyme is what? Which is it's really thermally labile and chemically labile. It's easy to dissociate, right? Can we make something like a heterogeneous catalysis behave something like enzymes, which is kind of dream in the heterogeneous catalysis in general, right? In the catalysis in general, right? That's what you're going to demonstrate here. So basically here is that a lot of recent activity in the so-called single atom catalyst. catalytic active site is single atom or single ions, right? That's what we try to do here. So the process that we modified from our 2008 Nature Material paper, which is we name is the wrap bake peel process. Basically, in short, first we pick up silica spheres, and then we did a sol gel chemistry of TiO2 sol gel chemistry on top of that, and then we introduce metal precursors such as cobalt, copper, or cobalt manganese, the salt on top of that, and then another layer of silica, and then cook it at high temperature. And then during this process, TiO2 sol gel become TiO2 anatase, most anatase TiO2. Simultaneously, that the metal ion, transition metal gets in and take it out the TiO2 substitution reaction occurs to make a single, and then we just etch silica with potassium hydroxide or like HF solutions, and we could get something like this. As shown here, is hollow titania. Actually, when you look at carefully, the skin is actually made of around 5 nanometer size TiO2, and that 5 nanometer size TiO2 is decorated with incorporated a single atom of copper. Because the copper and TiO2 are so similar in atomic number, it's not easy to differentiate. So we actually here is that the HRTM shows that the rhodium doped, rhodium single atom in TiO2 lattice. As we can see, clearly see that the bright rhodium, the spot there, which is single atom dispersed. And also we did XFs, demonstrate that there is no metal-metal bonding here. So showing that the metals are well atomically dispersed. That's what it demonstrates here. And then we did photocatalytic water splitting. Here we just use the half reactions, so basically half cell reactions. That's why actually we use the methanol as a whole scavenger that I have to mention that. So as shown here, one thing I want to notice is here is the white, right? There's nothing happens. And we turn it on a solar simulator. This is argon bubbling. There are small bubbles, as you can see from here. This is hydrogen generating here, right? And after reaction, now it becomes black, right? What happens actually that especially this is Samuel Mao's 2011 science paper is black titania. Black titania, it looks like black. So basically to make this kind of black titania, you have to cook it at high temperature in hydrogen and under high pressure, right? But here, as I demonstrated earlier, we simply shining the UV visible light here, solar simulator, we can in situ generate black titania, all right? And then it's black. That means it absorbs a whole bunch of light. Not only UV visible, but also even NIR light. That's why this gives a very high photocatalytic activity for water splitting here, which is actually nearly the same or better than standard PT like a TiO2 catalyst, PT nanoparticle and TiO2 catalyst. That's what it reported several years ago in the Nature Materials here. And then what we did a whole bunch of a DFT calculation by the DFT calculation by the Hyungjung Kim and Keist. As shown here, this is what's happening. So basically, there's something enzyme-like stuff process is occurring here. So initially, as you might know, photoactivation, right? So basically, the electron is transferred to the excited state, generating exciton, right? Electron hole pairs, right? And this excited electron is reduced nearby couple 2+ to couple 1+. But it doesn't stop there. That the couple 1+ changed surrounding electronic structure of TiO2. That's why it became black, as shown earlier, right? So basically, this is typically what's happening in the enzymatic actions, right? As a substrate approaches, right, the active site changes, right, adapt to the structure of the substrate. That's what the enzyme action is occurring. Similar stuff is working here, all right? That's what we have reported here. So and then more recently in collaboration with Suil in Digist, Korea, we also did the solid gas phase. CO2 reduction here, photocatalytic CO2 reduction to generate mostly methane. That's what it put in as a ES cover in the early last year. That's what we have reported. So final topic, I just 1 hour ago, I received the email the email confirmation from the editor. So I'm very happy to present this result. It's coming out today for tomorrow. Okay, actually Friday, this paper will come out in Nature Nanotechnology. So all of this kind of like a photocatalytic process, I mean, most of the people, most of the chemists work on to develop, as we did, as I just demonstrated, right? I just told you. Right? So most of the people interested in the developing new active photocatalyst, right? But there's a big issue here for the real commercialization, which is like a photocatalytic platform. So most of the powder form is like there's a lot of issues associated with the powder, right? Powder, to make it active, you have to keep on storing, right? But you cannot store seawater or liver water. It's impossible to keep on storing, right? That's impossible, right? You can do it storing in the laboratory, right? In the 500 ml, 1 liter, at large, you can do it. But you cannot store liver water or seawater even, right? So there's a lot of issues, right? And oftentimes there's back reaction. H2O2, H2 plus O2 goes back to water. That you don't want to happen, right? That's often happening in this kind of like a powder systems here. So here, my collaborator, Dae-Young Kim, gotta come up with a very exciting idea, because we've been working together for last 10 years on which I'm not going to talk about today, is actually we actually incorporate different kind of into rubber and the polymer, hydrogel. We incorporate different kind of nanostructural materials such as silver nanowire and quantum dot and that kind of stuff, right? We did that. So from that experience here, what we tried to do was we made a floatable photocatalytic platform here. So we actually use the one copper TiO2 single atom catalyst I just mentioned here. We incorporate this into the polymer plus polyurethane composite material, make it floatable on the water. And then using this kind of platform, we could produce a large continuous production of hydrogen. We could prevent back reaction. And also water is reactant, as you might know, can be continuous supplied from the bottom. And the back reactions are completely blocked here. That's why, as shown here, we made this kind of things. Again, we actually the standard PT TiO2 catalyst, and also our single atom copper in TiO2 catalyst too. And then, as shown here, compared to the immersed one, we intentionally just put into deep inside water, much higher activity when it float. All right, that's what I have demonstrated here. And also, to mimic the real situation, we actually intentionally put in some kind like a blue dye and the red dye and make it turbid, so that under that kind of conditions, still our photocatalytic platform is working great here. That's what I demonstrated. And also here again, we actually use that as a demonstration. We use methanol as a host scavenger here, but instead of using methanol, we can also use a PET, like the one you guys having here, right? That the PET as as a host scavenger. That we have demonstrated here. So hopefully this can be in the future commercialized for the large-scale production of hydrogen here. So I have to thank all of my students who did this work. We took this for 3 years. We couldn't take this picture, but then finally we could take it early April this year. And also, I always emphasize that I'm sorry. This is not in the ACS. This is Royal Society of Chemistry. I did interviews. I just emphasize here. So oh yeah, by the way, this is not Roger Federer. This is him Federer. All right. So if I'm not here, I might play tennis in the SNU, tennis court. Anyway, so as you might know, right now in the 21st century science, multidisciplinary, interdisciplinary collaboration is not optional; it's must. That's why we did a lot of collaboration for the last 20 years or so. I have to thank all of our excellent collaborators, and also like generous funding from IBS for the last 11 years. With that, thank you very much for your kind attention. Thank you.
Now the floor is open for questions and comments. Yep.
So I get to ask a question, right?
Yeah, I think you're allowed. Actually, the Andy Goworth is a professor at the UIUC chemistry, and I did the TA for him when he was assistant professor. That I have to.
So a lot of stuff, that one really beautiful. I sort of got lost after the first part, but so let me ask you a question about the first part. When you make platinum X nanoparticles, which you make a platinum shell on there, the shell is strained. Is the strain in your shell what is the strain in your shell?
Actually, the first part, I mean, and you're talking about the first part, right?
Yeah,
I
that's
mean,
correct.
IMPT alloy and the cobalt PT alloy nanoparticle. Shell, in this case, is we actually use start with the polydopamine coating.
Right.
That when you do heat treatment, that transforms to the end of the carbon shell. That is preventing that agglomeration during the fuel cell
Right. Got all that.
operation. Yeah.
But when you operate the fuel cell, it's platinum on the outside.
Oh yeah, that's.
The platinum is strained.
Oh
And
yeah,
What
yeah.
is the strain in your platinum? And have you figured out how to prevent then the platinum from dissolving during
Oh
long-term
yeah. Yes,
operation?
yeah, you're right. I mean, what Andy is asking you that actually that kind of cobalt PT alloy nanoparticle, typically platinum is coming out to the surface and give quite a lot of strain between that kind of like a transition matter, right? So how are we going to prevent? So that's one of there's a dual function of that end of the carbon turn out to be. Dual functions, which is the first one is it prevent interparticle agglomeration. Second thing is also preventing some kind of leaching out, not completely though. Okay, so we just published one paper in the ACS Nano. There's some editors here around us. So ACS Nano that we actually employ the Syria shares structures to exactly what we tried to figure out. Preventing carbon corrosion during the electric catalysis, and also preventing that kind of leaching out of the platinum. That we tried that. Yeah, that's what we reported last year in the ACS Nano. Yeah.
Okay, thank you.
And Yuri, yeah.
My question to a different part. You talk about black titanium.
Black titanium.
When you have this enzyme-like process with copper on the surface, do you get only the surface layer transformed, losing oxygen, reducing titanium, or the entire particle?
That's really important. That's why particle size is important. Particle size is important. As I mentioned earlier in the TM images, the overall structure is a hollow structure. But when you look at the shell, actually shell is actually made of the small, around 4 to 5 nanometer size, a tiny TiO2 nanoparticle. That means that pretty much all the copper, which is actually the substitute with the titanium, is actually exposed to the surface. That's why our catalyst, single atom catalyst, exhibit that high activity.
Thank you.
Any further question? To keep us on time, let's move on to the next talk. Please join me thanking Professor.
Thank you very much.
Kang Hyun
Thank you.
again. Okay, the next speaker, Professor Kwangjun Lee. And our next speaker is Professor Kwangjun Lee at National Center for Nanoscience and Technology. He is currently serving as an associate editor for Nano Letters. And today he will tell us about next generation of nano vaccines based on membrane vesicles.
All right.
Please
Thank you.
join me. Please welcome Professor Lee.
All right. Thank you very much, Chairperson. I'm so honored and pleased to speak to KCS and ACS people. I think just Professor Tan Zhen just talking about beautiful side of nanomaterials for catalytic reactions and also other applications for like material science. So today, just a little bit talking about our recent result about nano vaccines based on nanostructures. I think during the pandemic, everybody got a big feeling about how we can deal with the disease. And so today, just I show you some of our results. So this is my outline. So I first give a brief introduction about the field. What do we think about tumor vaccines and nanotechnology? So the vaccines we're talking about today is not the preventive, like for infected disease, but actually there are treatment options for chronic disease. And then after that, I focus on our recent results from the isosomes isolated from mammalian cells, move to the bacteria mammary vesicles. We use them as vaccines for treat the disease like different solid cancers. And then we move on a little bit about the hybrid mammary vesicles we made recently. So I think everybody I believe that cancer is a group of heterogeneous disease, and it's one of the toughest disease to treat. I think in the past, people spend a lot of efforts to try to understand what's going on for different tumors. And we do understand that cancers are the group of disease with the imbalance of our immune systems. And our immune systems really work very well to recognize and differentiate between our own and other non-self antigens. That can help to identify the malignant aging cells and get removal of them. But they still have chance, like malignant cells can escape about our immune attack and eventually grow up as a solid visible mass, tumor mass. And in the past decade, probably we all heard about that the major breakthrough in cancer treatment are immunotherapy based on the CAR T cells and also immune checkpoint blockade therapies. I think now more than about around 20–30% of various solid tumors, they respond very well to this immune treatment options. But still, the majority of tumor cells, especially the solid tumors, they really didn't respond very well to this two treatment. So we have another weapon now people really actively pursue for that. So this is, we call it vaccines. And tumor vaccines can be beneficial to the patients who do not respond very well to the other immune therapeutic options we mentioned, or other simply because we don't have the proper targets. So this slide highlights the difficulties why tumor can get through our immune systems, because tumor cells can express the antigens with lack of specificity and low immunogenicity, and also with some immune suppressive environment. So basically that block our current treatment efficacies. So with nanostructures, we're talking about the vaccine delivery systems or vehicles. We can enhance the antigen delivery and make a better presentation to antigen presenting cells. And then at the end, we can have better antigen specific T cell activities. That will be work very well for improved efficacy and specificity and long-term immune memory effects. Okay, although we know there are many different types about tumor vaccines, but from our chemistry point of view, there's only three major parts. Either they are tumor antigens, can be nucleic acids, can be peptides, or even tumor lysis. And also we have immune adjuvants. According to current proved immune adjuvants, there are only 6 types used for the preventive vaccines. But there are many different types under actively pursued by academia and the industry. And you can have a delivery vehicles or not. But when you have delivery vehicles, for example, liposomes or LNPs that can make our antigens even stronger or better to fight the disease. And I think we heard from the news, both BioNTech and Moderna, they pronounced that probably in the next few years, they will see personalized the mRNA vaccines for patients. Right. So that's a very brief summary about the field, probably since I think a lot of students are not in the field. So now I switch to what we done in the past, why we choose, say, nature existing materials for use as tumor vaccines. Actually, we done this by accident. I think back to 10 years ago, we studied nanomaterials interactions with our biological systems. And we found that when we give the mice like inhalation about iron oxide nanoparticles, what we found from the TEM. So if you look at here, we have a lot of membrane vesicles, looks like secreted from the donor cells, and they can interact with the downstream cells and even trigger more smaller membrane vesicles released. And then we purify this from the auxiliary lavage fluid. We figure out that after centrifugation, all of the membrane vesicles turn out to be the cup-shaped structures like this here. So basically, it's a high speed, make them like structure change, like our red blood cells. So at that time, we are very interested about this. We just want to study more about what this kind of vesicles called and what they do for our biological systems. So at that time, actually, we found out people already studied this kind of membrane vesicles from our immune systems, from red blood cells. They call them isosomes. Basically, that's the cell organism secreted by the cells. At that time, people don't understand what really the function is. But in our study, we found that our antigen-presenting cells can rapidly take up all of the membrane vesicles secreted into the blood, so that quickly remove them. So that's give membrane vesicles first give some hints that probably they can work as a signal molecules because they come up very quick and can give release in the blood and get move out the blood very quickly. And by various of studies, we found out that this APCs, after take up the membrane vesicles, taken either by two ways to activate our T cells. We call it direct or indirect, and they can switch the T cells towards what they call the Th1 polarization. And if the animals or the models we treated with iron oxide nanoparticles with existing inflammation conditions, and the mice probably get very severe inflammation. So this is just try to let us correlate it when in the bad weather. So when the patients with existing inflammation conditions can get bad to inhale something like the automobile's exhaust gas. Okay, so that's our first study. We figured out that actually the membrane vesicles are probably very important as signaling molecules. And then we just think about if we can use them to do some good things, right? At that time, I think US FDA just approved the first DC cell vaccines called Prevent. So we just did similar studies. We treat different tumor cells and purify their membrane vesicles, isosomes, and do the pulse trace with antigen-presenting cells in vitro, and then purify the membrane vessel from the DC cells, and then use them to immunize different mice. They're bearing different tumor burden. What we found that whatever tumor the vesicles coming from, they show similar structures as we saw before from the mice lung secreted these cup-shaped structures. And interestingly, we found that we can treat mice with the membrane vesicles. They bearing two different type of tumors antigens, and they can prevent both. And also, you see some of the case are even stronger effects, inhibition effects, and some the others not. But we found that if you have different type of tumors antigen together, you can actually enhance, so a sort of addictive or effects can increase the efficacies. So this is our first touch on the membrane vesicles. So we work with mammalian systems. At the same time, we're also thinking about if we can work with the bacteria secreted membrane vesicles, because everybody knows that bacteria are the most easy tools for us. We can do genetic modification, make large amount of production. We can use them to treat to give them to the animals, and because they have very advantage. For example, they also like, although the bacteria have the cell wall, but they can still like budding to produce a lot of these mammary vesicles, especially for the gram-negative bacteria. They can have a lot of these mammary vesicles, the small one. They can secrete it, use as the between the species to exchange information and materials. And also the size is about 30 to 100 nanometer. That's perfect size for lymph nodes targeting accumulation. So that's perfect as delivery vehicles, as vaccines. And also because we know bacteria have a lot of components in their membranes that containing the so-called pathogen-associated molecular patterns, PAMs in short. So that can trigger our immune system produce very strong effect against the invasions of the bacteria. We went to the literature to see how people use these mammary vesicles, and we found that people already use this pathogenic microbial mammary vesicles to treat infectious disease, especially for the one we call it here, Meningococcal. That's a big problem for some countries. And if you use this the bacterial secreted mammary vesicles, they can use as vaccines. But the problem is how you can link the known antigen to these mammary vesicles. So that's a current method. Either you can joint express on the membrane, you can make physical mixture, you can do chemical conjugation, and even though you can do encapsulation inside the mammary vesicles. So there's a major difference, a challenge we're talking about tumor, because tumor is very heterogeneous. You have a lot of mutations that make tumor cells very different. They can express many different antigens. So they are not like a pathogen. You only have one or two major antigens. So how you can link all of different antigens into the mammary vesicles, that's a challenge. So we can do the membrane to call the overexpression, let them have one on the surface, that's okay. But we learned something from the biological chemistry. They have some systems called plug and display systems. In this case, we have the catcher and tag systems. We can link the catcher to the membrane protein called the CLA that can be expressed on the surface of the mammary vesicles. On the other hand, we call that tag that can link with any known peptide or protein antigens. So with this case, we can mix them together in relatively physiological buffers. They can link together very efficiently. So with this case, we call them protein plug and play, and you can easily link many different peptide antigen on top of the membrane surface. So if you look at this figure, here we use gold nanoparticles, different size gold nanoparticles to show the bigger one shows one type of antigen, smaller one shows another one. So in this case, we can basically, eventually we can link up to 30, 20 different antigens together. And then, so next we just want to say, if we have, for example, dual tumor antigens, what we can do better than single. So in this case, we took the model antigen called OVA. We took two different peptides and immune animals to see if we have two antigens together, what's going on there. So we found out first, we found that the OMA system can simultaneously display two types of antigens. And also we see that when you have two types, they can increase either one or two, so can have a synergistic effects or addictive effects. And later on, we try a real mice known tumor antigens. This is called ADPGK. It's a real tumor antigens. We treat animals with MC38 colon cancer model. In this model, we found that even though we only have one known tumor antigen, they can generate very strong anti-tumor effects. See here that the tumor growth dramatically slowed, and also the survival increased dramatically. And this is also, if you compare our system with a previously known like single DNA strands, like the adjuvant, we see that our mammary vesicles better perform than that one. And then because we're talking about vaccines, they have to be long-term memory capacity. In this case, we did experiments. So we gave the animals 3 injections of the vaccines, and after 60 days, we see if the vaccine treatment can prevent tumor growth. Indeed, we found that if with the known, with the relevant, for example, the OAV as antigens, so you can see that can have very strong inhibit effects, killing effects. But if we take another one, unrelated, without the known tumor antigens that didn't work as a tumor vaccines, that tells is either it's antigen specific effects, or it needs to find out some new antigens for that type of tumors. So that's what show you the first case. We work with known peptide antigens. So people now interested about if you can deliver mRNA with memory vesicles. So we try to done this also using the similar strategies. So how we can link a known mRNA molecules to the memory vesicles? We go back to the literature. We found that in the nature we have many RNA binding proteins. We chose one called L7AE. That's a single unit they can bind to a specific RNA sequence called CD box. So meaning if we have the CD box link known mRNAs, you can have the L7AE on the surface. You can do the plug and play as we play with peptides, link that to the memory surface. And in this case, we found out, at least we can find out more than around 40% of cells, they can express the known mRNA antigens in the cells. Again, we studied in vitro innate immune activation. We found out all of these mRNA molecules, they can trigger so-called TLR 2, 4, 5 different subunits activation. And this innate immune activation can trigger later the later stage of T cells mediated T cell anti-tumor efficacies. And well, we also did the direct comparison. If you look at this picture, so this line is we use peptide as antigen, and this one we use mRNA as antigen. So we see that they basically can achieve similar anti-tumor effects, and the peptide can achieve the plateau a little bit early, and the mRNA can last a little bit longer. And again, we do the in vivo immune response measurement. We figure out that this also is a CD8 positive cytotoxic T cells mediated anti-tumor procedures. We measure this also in the real tumor antigen. We did set up a subcutaneous tumor model. In this case, we set up different groups. We just want to compare different efficacies with our systems. Later on, I think when we submit the paper to journal and they ask us if you can compare the currently known, well-known benchmark systems, for example, the liposome LNP systems, if you can do a similar or better job here. So you see, we did a liposome-based formulation and LNP did formulation. So in our studies, in terms of interferon gamma positive CD8 T cells, we can achieve a little bit similar effect levels and also similar anti-tumor effects. So this tells us that although LNP is well optimized systems, our system is newly established, but because we can assimilate innate immune system that can add on to a T cell anti-tumor immunity, so make these systems even better. So that's something worth to pursue future. All right, so now I show you a few cases how we work with peptide antigens, mRNA antigens. But all of them, we have to give the animals injection. But thinking about how we can change that to oral administration, there will be easier, more patient friendly. So, but problem is for oral delivery, you need bypass our harsh the digestive systems, You can have to get into the intestinal epicellular layers. That's quite challenging for most of the things. So in this case, we play with the bacteria used in our lab. We use E. coli as systems. We just force a known protein antigen under the promoter control of arabinos. That means we can let the bacteria produce a specific tumor antigen. And this antigen is not secreted in the local environment. Instead, they express into these memory vesicles. We also link another marker, Fc fragment of this protein, make it DC cell more friendly, interact this, they can increase the activation of DC cells. So with such case, we make a bacteria as a vaccine carrier, let them pass through our digestion tract and let them into the gut. They can secrete in vitro about the memory vesicles. That memory vesicle, because small size, they can pass through the epicellular barriers and then they activate the immune cells there. So this is in vitro, in situ anti-tumor vaccines. So when we did some experiments, I show you here. So when you use a luciferase label the bacteria, you can find that when you give orally, the bacteria pass through our intestinal tract very fast. Even though after a few hours, they get into the colon and cecum, right? And importantly, if we measure the memory vesicles in the tissue of the colon, we found that they co-localize with the DC cells. So that meaning they already pass through the barrier and also interact with the DC cells. And this is the colon cancer model. We want to see if use such oral systems really indeed inhibit the tumor growth. Such results have been summarized here. So we have different groups. So the last one is our experimental treatment groups, and compared to other poly-IC known adjuvants and with the antigen. So our system better perform than the previous known one. Yeah. More importantly, we want to answer the question people asked. The bacteria can be degraded. The fragment can be doing something, or the bacteria can secrete something. So we did important experiments here. So we set up 5 groups here. So control groups, and this is oral, give the memory vesicles and give the intracolonical injection of the fragments of bacteria. And this is our intracolonical injection of OMVs, memory vesicles, and this oral bacteria. We figure out only the last 2 groups show the therapeutic effects, meaning the intact bacteria can produce in situ memory vesicles as vaccines, or you can inject memory vesicles through the colon, give to the animals. So that strongly support our idea. So finally, we also evaluate long-term immune memory effects. So in this case, we give the animal 3 shots, like a few days, and then after 50 days, we see if the treatment can also protect the animals. So here we show you that indeed we show certain degree, not 100% inhibition of tumor growth. And if you look at the tumor lung metastasis, also see beautifully inhibit the tumor metastasis, but we still have some modules here, not 100%, but quite significant. Okay, so far I show you a couple of cases how we work with memory vesicles and try to set up some new type of vaccines. But in our current clinical scenarios, surgery is still the main treatment for solid tumors. But after treatment, the post-operational recurrence and metastasis is challenged. So how we can use our system combined with that to treat solid tumors? So in this case, we just mimic the surgery. We took a mice. They grow a big chunk of tumor burden, cut them off, and preserve the tumor mass and isolate their memories from there, leave a little bit, maybe 1% to 5% of tumor, let them grow back. So this type of things, we call them memory antigens. And this is, we also take E. coli and we digest their memory, the wall, bacterial wall, and get the protoplast, and also purify their memory here. So in this case, we got chemically similar lipids by layers. We can do some manipulation here. We can make the so-called hybrid memory vesicles. We have a nanoparticle called the PLGA as a core and coat with different type of memories. So here you can see from our TM, you can have either a tumor-derived or bacteria-derived or hybrid-derived. So they also similar size and see after on top of the core, you have a memory coating there. So very strongly, we found that whenever you have a little bit about a bacteria component, our antigen presenting cells get 100% activated. But we only have tumor-derived tissues or the memory vesicle, they didn't take any. So that's our immune system recognized the invasions. We took that similar strategies. And also in vitro, we found that our hybrid memory vesicles, they can stimulate DC cell maturation, and that can be translated into T cell, antigen-specific T cells activation. So compared to different controls, there are up to 10 times increase about T cell, CD8 T cells. And we then evaluate anti-tumor effects with different models, such as colon cancer, melanoma, and this is breast cancer models. So again, combined with the surgery, we cut the majority of the tumor burden and left over and let them grow back. We found out if without treatment, after a few days, the tumor grew back very fast. They can reach very big tumor burden. But with our hybrid memory vesicles, basically we can 100% prevent the tumor recurrence. So this is very promising and interesting. And also after 6 days, after all of the treated the mice, they're already tumor free. We give them a second challenge. Again, we see that all of the tumors get 100% inhibited. But you introduce another new tumor, that tumor grows very fast. So meaning this is T cell antigen specific immune therapy. So that's also highlight that really tumor preventive tumor vaccine is very challenging. We also try to figure out what are the major cellular players there. So we just try to delete all of the major immune cells like macrophages, neutrophils, T cells, B cells, macrophages, etc. At the end, we figure out that not only CD8 positive T cells very important, but NK cells very important. So this is quite different compared to the traditional anti-tumor vaccines. And we also direct compare with the known single pure MPLA adjuvant. We compare that, we found that our system better performed. So in the end, we figure out that collectively the enhanced antigen efficiency and multi-adjuvant and the vehicle together, they can do this better job. All right, that's something I present today, all of our recent studies. In summary, so we're talking about the tumor vaccines. Why are we talking about the future? Because now we already passed through the initial radiation-mediated tumor lysis, and in 2010, that's a DC cell-mediated the vaccines we call the Prevenge. That one also have been opened up the whole horizon of the field, but that one not very successful in clinic. Currently, the mainstream are using the sequencing methods. They sequence the patient samples to get out of the mutations information and predict what are the tumor new antigens, and then give them either peptides or mRNAs. So that's something current practice. Our memory vesicle system can combine with peptide mRNA system, and even we have some oral delivery systems that can be combined with the current systems. But for the other unknown tumor antigens, right? You don't know what are the real antigens are there. So we can use these hybrid memory systems, meaning so whenever you have a piece of these samples from the patients, you can use that as a materials to generate personalized tumor vaccines, right? And we sum up all of the details here in this protocol. We just want to, as graduate students can follow that, can make the field booming. Okay, so in terms of other biomolecule structures, the because the topic today is broad. Yeah, so we have peptides, nucleases, and memories together. So all of these things can actually they can be used as a vaccine delivery vehicles, other than only liposomes or LNPs. So finally, again, I just want to give my great thanks to the organizers, especially Jiaming, to organize this ACS and KCS meeting. Thank you very much for your attention. Thank you.
We'll take one question.
I use up a lot of time.
Can you,
Yeah, actually.
can you come out and use the standing mic?
Yeah.
Oh.
Somebody else? Go ahead. Is there anybody else? Oh, okay. If not. Oh, okay. I'm here. Senator, yeah, nice talk as usual. Senator Guangjun, one of the issues that, as you might know, the COVID-19 mRNA vaccine is successful
Yeah.
because it can be easily scaled up,
Yeah,
right?
yeah.
Because based on the lipid nanoparticle. As far as I remember, exosome and the vesicles, what you are using here is I don't think it's that easy for scale up. What do you think about that?
Okay.
And
Yeah.
also, in addition to that, probably you don't know the details about the Moderna or
Yeah.
BioNTech's cancer vaccine, right?
Yeah,
Is that also based on the lipid nanoparticle?
Yes, they all based on. So I think BioNTech and Moderna, they also use LNP-based delivery systems. LNP now is really optimized system. A lot of people are using that, but still that system is not perfect. So I think people say there are some side effects associated with different reasons, but one of that is some of them are empty, not without the loaded mRNAs. So you have to increase the content to have that better efficacy. So that's one reason. The other one is so you see our memory vesicles that can make the MR or peptides even stronger. So that can be integrated in the current systems. And for most of majority of tumor patients, you never know what their real new antigens are. So in that case, you cannot really use mRNA or peptides because you don't know who are the new antigens. In this case, our simple strategies, even though you don't know what they really are, that we can make individualized, personalized treatment options for them. And another advantage is very fast. You can make them in few days. The mRNAs or other based on the sequencing prediction stuff, they can take months, weeks. Yeah, just there's something worth to pursue. Yeah.
For the benefit of time, we'll move on to the next session.
Thank
Please
you.
join me thanking Professor Ni again.
Okay, this is Professor Song Joo-hang at MSc Department in Yonsei University. I serve as the Vice President of Foreign Affairs in KCS. I'm very happy to organize this ACS Publication Summit in our General Assembly with Professor Nam. Okay, let me chair the second session of this Publication Summit. We're going to have two speakers. The first one is the Professor Yuri Gogochi. He's the distinguished university professor and Charles T. and Lucien Bach professor of the MSc at Drexel University. Also, he served as a director of the Drexel Nanomaterials Institute. He made a principal contribution to the development of the materials for electrochemical capacitor and other energy storage materials. Especially, he discovered Maxen and the related materials. Also, he developed a lot of the carbide-based carbon material with tunable structures and porosity. He is recognized as a highly cited researcher in material science and chemistry field and the citation laureate by the Clarivate Analytics. He has served as an associate editor in the ACS Nano for 8 years. Currently, he also serves as the advisory board member in ACS Nano. Please join me welcome the Yuri Gogochi.
I guess I need to use a microphone, right? Yeah, to enable everyone to hear me. So what I'm going to do over the next 25, 30 minutes is to talk to you about a new, relatively new, but very large and quickly growing family of materials, which are called MXenes. Basically, those are two-dimensional carbides and nitrides, or can be oxycarbide if you replace some carbon with oxygen, or carbon nitrides if you have both carbon and nitrogen together, of earlier transition metals. So in these structures, you can see red atoms are transition metals, black dots connecting them, carbon, nitrogen, oxygen. They come in variety of structures, variety of flavors, colors, plasmonic colors. And this family of material has been attracting a lot of attention lately. Just to better explain for people who are not familiar with those of what they are sub-nanometer thin typically structures, few atoms thick. But what is important, most of them are very good metallic conductors with a high density of state at the Fermi level with a high concentration of carriers. Even graphene that is praised for its conductivity is zero bandgap semiconductor. So we have a material which has a conductive core due to the electrons of transition metals. However, their surface is terminated by oxygen, hydroxyl groups, amino group, halogens, chalcogens. So basically, you can get, for example, water dispersible metal or conducting clay if you want here. And this is a very unusual combination of properties for metallic materials. And as I mentioned, they come in a variety of different structures. The first one discovered was titanium 3C2. It was published in this paper, Advanced Material 2011. But in the next paper in ACS Nano, we announced this entire family of materials here. Michael Naguib was a PhD student working with Michel Barzoum, myself. He produced the first few MXenes. He is a professor at Tulane University right now. And the story continued now. How we make them? We take typically ceramic precursors, like, for example, MAX phases, which are layered carbides, nitride layers, separated by monatomic layer of, for example, aluminum, silicon, gallium, other elements. Keep in mind, there are strong metallic or covalent bonds depending on the element here. So you cannot just mechanically exfoliate them here. What you need to do, you need to etch away, remove this monatomic layer, for example, aluminum is shown here, titanium 3, aluminum C2. After you etch them away, now there are weak van der Waals or hydrogen bonds between the layers, and these layers can be easily separated into single layer flakes and dispersed in solution, because keep in mind, at least if we do etching, wet chemical etching, we get hydrophilic, easily dispersible material right away. There are many other techniques. For example, it's possible to use emulsion salt for some MXene, like nitrides. Wet chemical etching doesn't work very well. Will be different surface chemistries here. And since there are at least 150 MAX phases and a couple of dozens of other layered carbides, we can etch many of them. You can use DFT to simply calculate the bond strengths between MA element and X and M element. So, and again, I probably didn't mention it here. M stands for transition metal, X for carbon and nitrogen. So if I can break successfully MA bonds, if the bond energy is lower, bond is weaker basically than the max bond, we can selectively etch MAX phases, make MXenes here. So in this article, it was an editorial we wrote with my former postdoc, now professor at Purdue, Babak Nasuri here. We stated that MXenes are quickly rising here. And you see this article attracted quite a good attention after publication of ACS Nano. And you will see from the number of compositions, transition metals, blue M element. X at that time were only oxygen, fluorine, chlorine, OH. And variety of A elements that can be etched and variety of also elements you can intercalate. It's already very large number of materials. But just 2 years later, we wrote another editorial with Qin Huang here, showing that at least 10 of different surface termination were possible at that moment. A new subfamily with 5 layers of transition metal was added. And this growth continues very, very quickly. If you do a simple math and take a dozen of transition metal, carbon, nitrogen just alone, and 10 surface termination, you end up with at least 1,000 stoichiometric structures possible, of which actually only 50 have been synthesized, reported. But we can also consider solid solution. It becomes virtually infinite. And naturally, this very large family of materials has already attracted attention, at least over last 4 or 5 years. With, you can see, research is going on in 87 countries, on 6 continents. Korea, actually, number 3 after US and China in the number of publications here. Now, let's dig a little bit deeper into those type of structures. So what we can get? We can get materials with 2 layers of transition metal, like M2X structures, with various surface termination TX here. But what is important can be just single transition metal or a mixed random solid solution. And that will also be ordered in plane with alternating atomic columns of different elements. We can also get thicker structure with 3 layers of transition metal. Again, can be single M solid solution. And we can get out of plane ordering. When we have transition metal, like for example, moly or chromium on the surface, another transition metal, for example, titanium in the middle, like atomic sandwiches, out of plane ordering material things here. And we can get structures which will be twinned like this one with 5 layers of transition metal. So basically, we're really moving to the stage when we can do design of 2-dimensional material on demand with an enormous number of elements used, almost whole periodic system if you consider surface termination and intercalated atoms between the layers. And we can do it in all this variety of structures. And again, keep in mind, while number of layers of transition metal, whether they mixed randomly solid solution or separated out of plane or in plane order structures, or you etch out one of the element from in plane order structures and you get double vacancy, like a monatomic trenches going across the surface here. You can truly design material. And then you put on the surface this TX surface termination, further adding in the composition. So I think it really takes us to a new era of materials designed by demand. If you properly use computational science, guided by DFT, by machine learning, eventually artificial intelligence, we should really be able not just to take available materials, but really use materials by design on demand, knowing the properties. But keep in mind, MXenes are just one family in 2-dimensional world. We have graphene, we have dichalcogenides, we have oxide, hydroxides. Now we can actually combine all these materials and we have dielectric material like boron nitride, we have semiconducting, many chalcogenides, semi-metals, and now we have a large family of primarily metallic material. So we really can design new materials, new generation of materials and entire devices building from 2-dimensional bricks here. And of course, the system keeps expanding. Just a few months ago in this paper, we showed that we can replace carbon with oxygen up to 30% and get oxycarbide MXenes. And of course, it all affects the properties, and I won't have much time to look in detail. And this paper was published about a month and a half ago with a colleague from NIMTE and Sweden Linköping University, demonstrating how we can basically, again, on demand, substitute elements in max phases, creating very large number of structures. We can get gold inside, antimony, pretty much anything we want, single atomic layers, double atomic layers. And then basically using like emulsion salt, like atomic scissors, and then transform into MXenes, again, putting on the surface on demand whatever we want. Here is tellurium, selenium, and antimony. This was actually for the first time synthesized here. This one was shown by Dmitry Talapin, niobium, with tellurium, selenium being superconducting materials, things here. And it's really going on. And I know this is an ACS forum and it's wrong advertisement for science, but I wanted just to stress that just last month there were like 3 papers in Science addressing synthesis of new MXenes. And particularly this work Dmitry Talapin showed that it's possible just to take graphite and directly transform it into MXene, or you can do CVD of nitride MXene and carbide MXenes on the surface, making these beautiful structures that went on the cover here. So basically, the field is growing, add oxycarbide and carbonitride, you get a variety of materials. It's a great to have a very large number of materials with a large number of elements, large number of surface termination structures. But the question is here, okay, can we use those? Can we make them into something useful? One thing important to notice here is that MXene, majority of them are quite stable. For example, titanium 3C2 spent 6 months here in a dilute solution, 1 milligram per milliliter in a closed vial in water, and you see no changes in the Raman spectra, and there are some pin holes appeared after 10 months of storage in liquid, and you would not see them I marked them to make visible. IFM and tip enhanced Raman spectroscopy study showed that again, after 2 months, single flake in Petri dish don't really change at all. Things here. So we can use them. We can make flakes of different sizes. For example, this is a paper published end of last year in ACS Nano, shows about 20, actually we can go up to 40 micron flakes of titanium 3C2. Those are single layer flakes on a silicon wafer. Those are uniformly colored single layer. Those are a couple of double layer flakes. This one you see folded, things here. Has anyone ever seen moly disulfide, oxide, any other flake by graphene or reduced graphene oxide produced from solution in that sizes? I've never seen any. And the main reason is here. Those are others not strong enough to survive just vibration. They break apart. This is very high mechanical properties of MXene inherited from bulk transition metal carbide nitride that allow us to make this large crystal from solution in water. Moreover, there are many different nanomaterials which are produced in nano quantities. Since we make these materials by wet chemical methods, somewhat akin to graphene oxide, we can make them in any quantities here. So we have these reactors designed for us by Materials Center, Ukraine, and you see 100 grams of titanium carbide MXene coming from a single batch. This atomic sandwich, moly, 2 titanium C2 MXene, 40 grams produced by undergraduate student, and here one of my students is holding 1 kilogram of MXene produced within 24 hours in the lab. So we can make this in kilogram quantity, even in laboratory things here. And what is important, if you do wet chemical etching and bulk scalable synthesis, you can produce not only almost unlimited amount of material, it just depends how much ceramic powder you dump into acidic solution. But you can also easily disperse it because MXene flakes have a negative charge on the surface. Zeta potential is typically below minus 30 millivolt at close to neutral pH. So if you put them in solution, they will just simply disperse. You see different MXene give you different colors because they have plasmonic colors. So we take ceramic powder, etch it, delaminate it, make colloidal solution of 2-dimensional flakes. If you have a colloidal solution of 2-dimensional flakes, we can handle it exactly the same ways as a graphene oxide or clay or anything else soluble in water. And you will see, for example, this is dilute solution. We use it for inkjet printing, for example. This is viscose liquid crystalline solution. We use it for drawing fibers or ductile braid processing of the material. And you don't need any additives. It's just solution in water. So unlike carbon nanotubes, graphene, you don't need to add surfactant. Unlike graphene oxide, you don't need to reduce it to make it conductive and useful afterwards here. It's conducting right away. And pretty much you can get any concentration from slurry to very dilute solutions for making like assembly monolayer film, something like this here. And you see this what kilogram of MXene was transferred, for example, here to about 160 liter of colloidal solution of delaminated, mainly single layer with some double, triple layer flakes here. So it's a processable. If you have a colloidal solution of particles in water, again, no binder, no surfactant, you don't need to burn anything out. You can use any known technique. There is nothing new here to process it. 2D printing, 3D printing, you name it here. And what is also important, there are many exotic materials. most of MXenes are produced from abundant element. Take typical titanium with carbon, nitrogen, oxygen, fluorine, chlorine terminations, and you get just in this system a whole bunch of materials made of very abundant elements. So again, it shows scalability and potential use in application where large volume of materials are required. It's all great. We have an enormous family of materials emerging with almost infinite number of compositions possible. We make them in a scalable way, large quantities from abundant elements. It's all great, only in one case. If these materials bring something to the table that other available material can't. If you can do the same thing with clay or graphene, there is no point to look for something else. So what do MXenes bring to the table? First of all, this combination of metallic conductivity with hydrophilicity, surface terminations. No other material, to the best of my knowledge, offers something like that. Moreover, this multi-layer film of titanium 3C2, and actually we go well over 20,000 Siemens per centimeter, it hasn't been published yet nowadays here, is about 10 times higher in conductivity compared to reducing graphene oxide, that you'll have to make a graphene oxide film and after that reduce it at elevated temperatures in here. But they offer variety of plasmonic colors with plasmons ranging from UV to near infrared range, providing, for example, from infrared stealth to very high emissivity things here. The dispersible water is a process I mentioned. They're very efficient light to heat converters. You can use them from photothermal therapy to water evaporation desalination or photoelectrocatalysis or photocatalysis because of these properties and other. But what is the most important thing here? All their properties are highly tunable. So we don't have one material, one element things here. We have materials that can change the property. And we know we confirm, for example, take single titanium 3C2 flake and you will see this linear IV curve here. But we can potentially open the band gap, terminating the surface of thinner MXenes with oxygen, for example, which will pull 2 electrons away from each transition metal. We can control surface termination, number of layers, transition elements, carbon, nitrogen, oxygen, and the lattice. But moreover, we can actually intercalate ions and molecules, or we can covalently bond molecules to the surface without killing conductivity of those materials here, which is again very important because if you get oxygen OH on surface of graphene, conductivity drops by 6 orders of magnitude. And it doesn't have the case here. So you can actually chemically change the material. And property change is significant. It may be difficult to see from the back, but you will see here, for example, for hydroxyl terminated MXene, the work function is close to 2 electron volt. You change termination by heating it up to about 300 degrees C to oxygen, and it goes up to 6 electron volt. So we work with colleagues at SNU, for example, on using this material for electron transport layer, tuning work function to match organic for LEDs, OLEDs, and other devices here. And you can see all this variety of plasmonic colors. Just to mention, for example, titanium 2C is red in solution, green in reflection. Just like gold nanoparticle, with the only difference, there is no gold. Because surface plasmon is absorption peak is about 550 nanometers. You add 1 layer of titanium carbon extra, titanium 3C2 with the same oxygen H termination, and it becomes green in solution and kind of a purple in reflection because the peak shifts to about 780 nanometers here. And of course, by changing surface termination, you can reversibly shift it within 100 to 200 nanometers here, making electrochromic materials or again, actively controlling the properties. And this is again the key. We get now materials which are metallic, but metals with a tunable Fermi level, because changing surface termination, you can do it in situ by applying, for example, 0.5 volt potential in acidic solution, protonating and deprotonating the surface. And you can significantly change the density of state, oxidation state of material. So we get tunable metals. Can be done ex situ before, permanently, and can be done in situ. For example, we showed recently, I'm not going to show you today, tunable electromagnetic interference shielding, active modulation, which has never been done before this way here. And there are many applications based on this here. For example, we work in my group a lot on electrochemical energy storage, and everyone is talking about nowadays a high pressure, high energy, high power battery, something that you can charge, say, your Tesla car within 5 to 10 minutes. What do you need for this? You need to eliminate bulk diffusion. As long as you have bulk diffusion, it will never be possible to move ions quickly. Solid state diffusion is slow. You need to have redox process, because without redox, you will end up always with double air capacity and low amount of energy stored. And one thing that most people forget, you need to have high electronic conductivity. What does it mean, fast charging? It's very simple. You run a high current through your device. What happens when you run a high current through a poor conductor? Joule heating. And this is why cars have such sophisticated, expensive, bulky cooling systems here. And it also means a lot of losses here. MXene can potentially offer all of this. They're two-dimensional materials. They have a redox active, moly, vanadium, niobium, other elements on the surface. There are channels for transport of ion. Electronic conductivity exceeds all materials used in a battery technology today. So we believe there are many, many opportunities there. And I don't have much time. How much I have? 5 minutes? 5. Okay. Just one example, work coming actually from Korea, from collaboration with Chungmin Koo, Sunjun Kim, and other things here. 5 years ago, we published a paper showing that, actually time flies, 7 years ago, that titanium-32 actually being 2 orders of magnitude less conducting than copper aluminum lines up in thickness, in basically shielding. And EMI shielding of 50 decibel means 99.999% all the electromagnetic wave will be shielded in this band here. So it's behaved the same as aluminum copper. But unlike aluminum copper, we can easily, by just spreading and drying film, make thin micrometer freestanding film with mechanical strength of aluminum foil, about 15-20,000 Siemens per centimeter conductivity. This paper, within 5 years, became the most cited paper in the entire EMI shielding field ever. And in 2020, we published another paper showing actually that titanium carbon nitride MXene outperforms with conductivity even an order of magnitude lower than titanium-32, outperforms at the same thickness all the metals in shielding here. And the physics of the process is still very poorly understood. It's a clear interaction with individual flakes. But this is X-band. We have millimeter-long waves strongly interacting with nanometer thin, few microns wide flakes. Physics, open questions to address here. And we showed that other MXenes, for example, vanadium carbide MXene. So this is about reflection largely, like metals reflecting all the waves, protecting all the electronic devices around. But vanadium carbide MXene can actually absorb electromagnetic waves when dispersed in the matrix at a very, very small loading, like about 2% giving performance here. So just very early stage, it outperforms all other known materials in that field. And again, we have a variety of different MXenes. We can make them into film. We can make basically a lot of applications here. There are many other emerging applications I won't have time to talk about. Hussam Al-Sharif announced the field of maxitronics in this ACS publication a couple of years ago. And very recently, months ago, they published like a roadmap. So US brought this CHIPS program, competing electronic with China. In my opinion, we should not compete. We should leapfrog, designing new wearable, printable electronics by using all this variety of materials, two-dimensional, zero-dimensional, one-dimensional building blocks here. Go ahead, leapfrog current technologies here. And of course, there are many other applications based on properties of materials I have not talked today, ranging from desalination, gas separation membranes, to antennas, again, tunable antennas, tunable shielding. You can modulate it electrochemically when you put ions in between. So again, modulated metals. Sensing, gas sensing. First work was published in ACS Nano with Sunjun Kim when he was still at KCS here. Optoelectronic, I mentioned a little bit work on OLEDs, plasmonic photonics here, medical application, biocompatible material, for photothermal therapy to implantable brain electrodes to absorption of urea for wearable kidney here. And it's slowly getting towards commercial applications, interaction with industry. There is a Maxine Association bringing together industry and academia. And of course, there are tons of unsolved problems and open questions. From developing better CVD, PVD technologies to checking all the prediction on tunable superconductivity, chemical tunable superconductivity, two-dimensional ferromagnetism, antiferromagnetism, topological insulator, and so on and so forth here. And of course, we still don't know which of all these possible numerous applications already being explored will be a winner and killer application at the end of the day here. And if you're interested in looking into this, in this, it's already getting a little bit dated, but in this editorial, we published a list of challenges in Maxine field. And this was actually challenges that community voted at the third international conference Maxine in China. Few of them actually have been addressed in the meantime, but there are many open. If you're looking for opportunities, check this editorial and find the problem to work on. And this is my very last slide. Of course, all this work was done by outstanding students, postdocs, visiting scientists in my group. There are always some Korean researchers, of course, participating, and many collaborators. This is a small subset of people whose work I showed today. We're also collaborating with Professor Wang, Yonsei University, and many others who are not mentioned here. And I fully agree with first speaker who told that pretty much it's all collaborative work here. It's a must, it's a requirement nowadays, because solving this interdisciplinary program, working with so many different materials, so many applications is impossible if you don't collaborate. Thank you very much for your attention.
Okay, thank you very much for your very inspiring talk. Now this paper is open for question or comment. Maybe I can invite one short question. Do you have any? Okay, over there. Could you go to the micro instead of behind?
Thank you very much. It was a very interesting talk. And my question is, I believe that the properties change depending on the composition of elements. But what did you say the thermal stability in general, for example, mixins of carbon nitrides?
In vacuum, typically they are stable till about 650 to 900 degrees C, depending on Maxine. At high temperatures, start losing functional groups, surface mobility of transition metal atom increases, and then transform to like a nanocrystalline, like a still two-dimensional almost here sheets, but they have tiny nanocrystals here. In air, maximum oxidation stability we observe was up to about 450 degrees C. So they can oxidize, the carbides, nitrides, they can potentially oxidize, and fewer defects, larger flake size, dense as the film, high is oxidation resistance.
Okay, thank you very much. Due to the time limitation, I have to move to the next section. Please join me in thanking the speaker. Our second speaker is Professor Il-Du Kim. He is the chair professor of the MSc Department and KAIST. Also, he served as a director of Membrane Innovation Center for KAIST Institute. He devoted to the development of the novel method of the synthesizing various organic, inorganic nanofiber materials can be applied for chemosensor and medical application and energy application. He published a lot, also especially some of patent successfully the license to the industry. Currently, he served as an associate editor in ACS Nano. Please welcome the speaker.
Thanks for kind introduction, Professor Huang. And also, I would like to thank the conference organizer and also Professor Jin Nam for kind invitation. And today I would like to introduce the multi-dimensional the nanomaterials, especially optimized for chemical sensing application. Before introduction of today's talk, then I would like to briefly introduce our lab. Our lab name is Advanced Nanomaterials and Energy Lab. That we exploring various type of nanomaterials synthesis. Then we apply this material for sense application and also the energy storage application. I joined KAIST 2011. Then our lab has been well established on the 2015, then from 2016, we published about 30 article, and there is a very interesting spike, the 2018, then also we expect another spike, the 2023 or the 2024. So what happened, the 2018? So I was a sabbatical year in UC Irvine, so with the Professor Reginald panel group, then I'm not that sure, actually. Then you can see here, these are former my graduate student, then Reggie is very tall. Then also, now I'm the sabbatical year at the UC San Diego, the Professor Joseph Wang's group, then starting from this January. So actually, I'm from the United States here. Okay, so today I would like to discuss 4 different topics. The first topic is the electrosponge membrane, the nanofiber alignment. Then I would like to introduce conducting nanofibers, polymer nanofibers, and metal oxide nanofibers. Then I will discuss on the chemical registers using the one-dimensional the nanofiber. Then also, the catalyst decoration is very important. Previously, the Professor Hyun-taek Kwan mentioned the activity of catalyst is very important. Then also the mechanical stability of this catalyst is also important. So I would like to mention on this X-solution is a new catalyst design approach. Then finally, if time is available, then I also discuss on the 3D dimensional readable colorimetric sensors. Electrospinning is a very well-reputed technology for producing organic and inorganic nanofibers. So highly viscous polymer, once ejected through the nozzle, they are collected on ground substrate. In this case, we always see the randomly entangled nanofibers, non-woven fibers. So we invented this insulating block guiding electric field. Then after that, you can see the aligning of this fiber, the uniaxial direction back and forth. Then by rotating the substrate, the 90 degree, then we can get cross-aligned the nanofibers. So randomly oriented nanofibers, the aligned nanofibers, by moving the stage, the cross-aligned nanofiber can be easily synthesized. Of course, it's not 100% perfect. But compared to these randomly entangled fibers, you can see quasi-aligned fiber. Actually, this is very important in terms of porosity control. Then also we can increase the nanofiber density. Then also we can fastly writing this fiber, cross-aligned fiber, we can control the angle. Then by simple collection of this membrane, we can prepare the cross-aligned nanofiber. So how we can extend this fiber for specific application? The first example is antivirus filter. So we use the violacine. Violacine is a well-known antibacterial material. So we directly embed this violacine dye within the electrosponge fiber, then cross-aligned fiber provide much improved the filtration efficiency toward influenza virus and also human coronavirus. Another interesting approach is the PTFE. So as we know, this Teflon is not well dissolved in polar solvent. So that's why we use the PEO, is a matrix fiber. Then we embedded this PTFE in the nanoparticle. Then after preparing this composite nanofiber, then high temperature heating, we burn out this PEO matrix. Then after then, this PTFE particle fused together, then forming this cross-aligned PTFE solid electrolyte. So we successfully demonstrate the PEM fuel cell application using this PTFE, the solid electrolyte. Another interesting approach is the hollow, the ceramic tube. So we first prepare the PEO, cross-aligned nanofiber, then direct coating of ALD, aluminum oxide, then removal of the core fiber. After then, you can see here the cross-aligned, the hollow, the aluminum oxide, the tubular structure. Then we combine this tubular structure, the membrane, with the elastomer. Then before stretching, it is transparent. After stretching, it is opaque. So optically tunable, the membrane can be achieved using this approach. Another interesting approach is the carbon fibers. So we prepare the cross-aligned carbon fibers. So we expect much improved the high density using this type of the carbon nanofiber. Then you can see here catalytic particle decoration on this fiber. 2018, the Professor Liang-Bin Hu from the Maryland University, he successfully demonstrate the carbon thermal shock. So simply based on the Joule heating. So you can see when we apply bias temperature increase almost up to the 2000 degrees C, then very fast quenching. This is just the millisecond level, right? So 50 millisecond, very fast quenching, then we can get this high entropy, the nanoparticle. So you can see here the 5-element, 6-element, and 7-element multi-elemental particle can be easily synthesized using this Joule heating. So also we established this Joule heating setup. So you can see here, this is an electrospun, the carbonized fiber. So when we applied bias, temperature increase almost up to 1,500 degrees C. So we try to extend this membrane. So as you can see here, this is almost a 10 by 10, the carbon nanofiber membrane, 2 centimeter, 5 centimeter. Then also we can control the pulse, the number, and also duration. Then controlling this processing parameter, we can precisely control the particle size and also the density of this particle. So if you are interested, please refer the perspective, the multi-elemental, the nanoparticle. Then this is A4 size carbon nanofibers by simply the drop coating of the precursors. 2 precursors, then you can see here the bimetal catalyst, and 3 precursors, ternary and quaternary. The small particle can be easily synthesized. We can explore a variety of the multi-elemental particles for using this Joule heating approach. Okay, so this is another example. So we prepare high density the nanofiber yarn. Then by Joule heating, also we can prepare very tiny high density the nanoparticle. So gold nanoparticle about 10 nanometer, based on the Joule heating. Then we additionally use the gold particle decorated high density yarn as a seed mediated growth. Then you can see here the grape shaped particle can be synthesized. So it has a gap. So we use this material for the source application. Another interesting approach is catalyst designing, because the most case catalysts are easily detached from the substrate during the long-term cycling. So we decorated the very thin, ultra thin, the oxide layer by using the double step, the Joule heating. So first one is a carbon nanofiber, the Joule heating. Then we prepare 5 nanometer titanium oxide overlayer. After second Joule heating, precursor coating, then you can see very tiny small particle tightly anchored or buried in this oxide layer, providing much higher the long cycling stability. Then also you can see here the polymer nanofiber synthesis is very easy. So you can see the biodegradable, the polymer fiber, PB80 fiber, and also the PHA fiber. We successfully apply this polymer fiber for filter application. Then another interesting approach is the metal oxide. I'm the ceramic background. So we can use this polymer fiber as a matrix. So sacrificial template, then precursor, embedding, dispersion, the fiber synthesis, and high temperature calcination in air atmosphere, we can burn out this polymer matrix. Then inside the metal sulfur oxidized, then we can prepare very thin the metal oxide layers. Actually, this is aluminum oxide, about 10 micrometer thick aluminum oxide. So now we utilize this ultra thin, the nanofiber, for solid state electrolyte fabrication. Okay, so I'm very interested in the industrial commercialization. So this is a mass production line, all homemade. So we spent almost 4 years for this 35 centimeter width roll-to-roll machine. And this is a 1.2 meter, the width, the roll-to-roll machine. Then also you can see multi-nodule system moving the back and forth, so we can cover the large area. Okay, so let me move on the chemical registers using one-dimensional the nanofibers. Actually, for sensor application, high surface area is very important. And also high porosity is very important because this porosity can facilitate the gas diffusion and penetration for enhanced gas reaction. So you can see here the nanofibers with abundant pores, then also catalyzed decorated nanofibers, then also high surface area nanofiber by controlling the phase miscibility. If phase immiscible case, so polymer rich domain and precursor rich domain, this polymer rich domain after calcination converted to the pores, and precursor rich domain converted to the oxide, so we can prepare highly porous the nanofiber. Then also we can intentionally the embedding this polystyrene bead for creation of open pores inside of the fibers. The main beauty of this electrospinning, we can explore a variety of the metal oxide because it is a solution-based process. We can mix any type of the metal salt, then forming binary, ternary, and quaternary oxide can be synthesized using this approach. So we prepare semi-conducting the metal oxide nanofiber and simply chopping, then ultrasonication or the grinding, we prepare ink and drop coating on sensor substrate. We measure the chemical sensing property. So gas response is very simple. So N-type, the semi-conducting material, we first measure the air resistance. Then after exposed to the reducing gas, then you can see the resistance decrease when exposed to the fresh air again, it returns back to the original state. So let's compare commercialized gas sensors, and this is the electrospun, the metal oxide fiber, high surface area, high porosity. Then in the case of the catalyst decoration, in this case, densely packed the film, the inside catalysts are not active, but nanofiber case, catalysts are entirely exposed to target gas. So we expect the low concentration gas sensing property much improved. So we have explored the various type of the catalyst design because without aggregation, catalyst decoration is very important in sensor application. So just one example, so we use apoperatin. So hollow protein, so inner cavity size is 8 nanometer, the shell size is 12 nanometer, so we infiltrate the metal precursors and simple reduction process, we can synthesize very tiny the nanoparticle, about the 2 nanometer scale, then still surrounded by this protein shell. So never aggregated within the electrospinning solution. Then also we can infiltrate the 2 precursors at the same time, then simple reduction, that means we can prepare alloy based type, the catalyst material. Then another interesting approach is the co-catalyst. So this is a platinum catalyst and oxide catalyst. Co-catalyst provide much improved the hydrogen sulfide sensing property. Okay, so let me move on to the third topic, the using the MOF. So in chemistry, everybody knows what MOF is, but I'm ceramic, so the 2016, I didn't know what MOF is. One of our graduate students introduced, let's use this MOF for sensor application. So we can use the conducting MOF, and this is a molecular sieving layer. So we combine this MOF with the electrospun, the nanofibers. So I'm very proud of the first MOF paper, because this JOX paper is our first group, the JOX paper, then also our first paper in our department. So then we can simply consider this molecular sieving layer, because this GIF8 has a pore size 0.34 nanometer, and hydrogen, the kinetic diameter is a 0.289 nanometer. That means the below one is the palladium, the nanowire, which is a well-known hydrogen sensing material. We directly overcoat this GIF8 on top of it, then we can interfering gases can be filtered. So we can see much improved hydrogen sensing property. Another interesting approach is a high density nanofiber ion. So this is also homemade system. So one side is a positively charged nanofiber ejected. The other side is a negatively charged fiber. The ejected, they are collected. This is the fast rotating drum. After winding, we can get high density the nanofiber ion. So very increased surface area. So ion diameter, this is about 0.26 millimeter, 260 micrometer. Very small, but a lot of fibers, more than 100,000 of fibers are entangled together. So we can utilize this ion as a flexible sensing platform. Simply, we coat ITO on top of it, conformal coating as a sensing layer. Then after then, we additionally grow the ZIF8 for gas separation, gas filtration. So you can see here, this is a pristine ITO coated ion, and ZIF8 1 hour and 2 hour and 4 hour growth. After then, the 2 hour growth, we found the conformal encapsulation of ZIF8 on ITO. Then as you can see, without ZIF8, there is no specific gas reaction, but with ZIF8, we found very improved, much improved the NO, the selective response using this multifunctional ZIF8, the filter, the nanofiber. So you can see this is just a platform, right? So we can utilize this nanofiber ion, then we can combine many different type of the material, then also we can combine the various type of the GIF or MOF material for selective filtration. Another interesting approach is exsolution. So exsolution, as you can see here, before exsolution, it is clean. But after exsolution, very tiny catalytic particles are absorbed from the lattice to the surface. So lattice doped element, the reducing gas atmosphere annealing, it is absorbed. So that's why we need high temperature reducing atmosphere annealing for this exsolution. So most case, this exsolution occurred in the perovskite material. B-site doped element is easily absorbed. The main beauty, main advantage of this exsolution, these catalytic particles are socketed, tightly anchored on support material, so we can expect the high mechanical stability. So if you are further interested, please refer this ACS Nano, the review article on this exsolution catalyst. As I told you, this exsolution behavior is reversible, the reducing atmosphere, then oxidizing atmosphere, reducing atmosphere, oxidizing atmosphere, the annealing, that we can see exsolution particle then formation. As I told you, this exsolution is the widely studied in this perovskite-based material. Then we successfully demonstrate the two-dimensional, the tungsten oxide, the sheet structure, low temperature exsolution, just 300, 400, 500, relatively low temperature is enough to making this extrude particle on this nanostructured material. Then also recently we successfully demonstrate the exsolution, the behavior on this MOF-based material, the zinc oxide based material, and bimetal catalysts are easily extruded from the zinc oxide lattice. Okay. Then I told you for this exsolution, the high temperature reducing atmosphere annealing is required, right? So it takes so long time. For example, 6 hours and sometimes it's 1 day, right? So here most people are Korean, so we should be faster. So that's why we introduced the IPL, so optical sintering, using the xenon flash lamp. So just to use, the support is oxide. This is a tin oxide SNO2, the 2D sheet. Then you can see this is a xenon flash lamp, just a simple exposure. So 20 millisecond exposure is enough to increase temperature up to 1,700 degrees C, okay? Just a simple exposure, 20 milliseconds, very short time IPL exposure, then you can see multi-elemental nanoparticle simply synthesized. We just simply the drop coating of these metal precursors and simple exposure, the IPL. Then we measure the temperature is almost 1,700 degrees C. Then we also extended this IPL on this tungsten oxide nanofiber, platinum, rhodium, iridium, tungsten oxide nanofiber IPL exposure, and small particles are extruded from the lattice doped element to the outer surface. Then we can see here very high the sensing performance. Before exsolution, after exsolution, sensing properties dramatically improve. Then you can see long-term stability of this extruded the particle. And recently, we extended this single atom catalyst synthesis on this plasma shock synthesis. So for example, graphene oxide, then melanin is an endoping site. Then metal precursors, any type of metal precursors can be synthesized. Just a simple exposure, one-time IPL exposure, then we could see the formation of abundant single atom catalyst on the reduced graphene oxide. Okay, let me move on to the last topic. I'm very interested in these eye-readable colorimetric sensors in terms of industrial commercialization. So first one is paper, just a paper, is the litmus paper, and second one is the membrane type, the commercialized colorimetric sensors. Is third one is the nanofiber based colorimetric sensors. Let's compare the surface area. It's much high, right? Then also open pores, so we expect the gas diffusion into the inner layer. Entire surface actively participate in the color changing. Okay, so this is a example, dye decorated the nanofiber. This is a gas gun riding the 20 ppm gas exposure. Then you can see very fast color changing. Then also 1 ppm, the resolution can be achieved. Then this is a HCl vapor reaction. This is ammonia, the vapor reaction. Before ammonia exposure is yellow. Then after ammonia exposure, you can see the color changing from yellow to white. Then also we can utilize as a backbone, as I told you, we can use the high density nanofiber yarn, the dye decorated yarn, then we prepare the stitching pattern, H2S, KCS. After exposure, you can see the color change. So this is ammonia vapor reaction. So you can see very fast color changing reaction. Another interesting approach, not just gas reaction. So this is a thermochromic dye, the loaded nanofiber, then 39 the degree C, then temperature, then you can see the color change, right? So from black to white and blue to white. So we can measure body temperature using this type, thermochromic dye decorated the nanofiber approach. Okay, so as I mentioned, we are very interested in industrial commercialization. So we can utilize this nanofiber for leak detection in semiconducting production line. So simply we can wrap this nanofiber, then close the system, then outside the camera can monitor this color change. If there is a color change, then we can see the signal transfer to this kiosk. So people original state is clean. Then after gas reaction, we can see the gas alarm. So now this system is installed in the Samsung, the semiconductor production line. So now it's under the evaluation. Okay, so take-home message, then I emphasize catalyst design, then also gas separation is also very important. In the viewpoint of we explore various type of the material, conducting MOF material, and molecular sieving layer is very important. As a new, the sintering approach, I introduced the Joule heating and IPL. Then as a new sensing platform, I introduced the nanofiber yarn. Okay, so I would like to thank all our group members and also all collaborators, and also I'm very welcome the impactful collaboration. Thank you very much for your attention.
Okay, thank you very much. Very inspiring talk. Now this paper is open for question or comment. Do you have any question?
Yeah, I do very nice work. So I have one question about the selectivity. So now you demonstration of sensing. Take a last example, right? You demonstration this kind of ammonia gas sensing. Now the selectivity. How's the selectivity there? Sensitivity, no problem. How about selectivity?
Right, so that's a very good point. The gas selectivity is always issue, but we can, for example, then hydrogen sulfide gases. These sulfur species can have a certain reaction with a specific element. For example, copper sulfide, the nickel sulfide. So once we have the very tiny copper particle or nickel particle, we can provide certain selectivity. So now, currently, this gas always react the many different catalytic particles. So that's why we can prepare the array. So pore sensing array, if there are four gases are mixed together, then we need 4 sensors. Based on this sensing array, that we can calculate the principal component analysis. So then we can provide additional selectivity.
Is there any other question?
Thank you very much for your nice presentation. Actually, I also worked with like carbon fiber based on polyacrylonitrile. So my question is that in case of your nanofiber, so you like dope to different kind of metal combinations. Are those nanofibers hydrophilic or hydrophobic? Like
Usually, that's a good question. So usually the carbonized fiber is always hydrophobic. So then we need for catalytic decoration, we need hydrophilic surface. So simple oxygen plasma, then we can provide hydrophilic surface, then drop coating of the precursor solution is easily well attached on the fiber. Then also we can use the simple acidic treatment to convert hydrophobic to hydrophilic. It's not a problem.
is carbon nanofiber itself has catalytic property or not?
No. So we additionally decorate the catalytic particle on this carbon fiber.
Okay, thank you very much.
Any other question? If you do not any, I'd like to wrap up this session. Please, thanks to the speaker. Actually, we have the 30-minute coffee break, but the begin the coffee break, we're gonna have the plaque awarding ceremony. So please be seated.
All right, everybody. I would like to open the second session of the ACS, the symposium, the KCS symposium for the ACS summit. So I'm pretty happy to be in charge of this session. And my name is Hye Byun, working at KCS Chemistry. And the two speakers are ready for this session. So I'm really happy to introduce the first speaker, Professor Jennifer Dion, in material science and engineering of the Stanford University. And she also served as the associate editor of the Nanoletters. So yes, let's kindly welcome to the Professor Jennifer Dion.
Great. Thank you so much for the opportunity to speak here. I'd especially like to thank the organizers, Joamin and Songju. It's amazing to be here in Korea. Yesterday, I was at Seoul National University and mentioned I love Korean food, love K-pop. So it's great to be able to spend the week here. It's my first time in Korea and excited to do science Gangnam style. Since the theme of the talk is post-pandemic transformations, I wanted to share some of the work that my lab has been conducting in the last few years, taking many of the insights that you've heard from previous speakers in terms of kind of new materials and new methods, and applying them to pathogen detection, in particular bacterial infections. And before I get into the science that our work has really focused on, I just want to share with you the broad theme of my lab, which is developing new methods both to detect molecules and to direct their transformations. And we heard some beautiful talks earlier today about bimetallic, metallic nanoparticles for catalysis. My lab has spent the last decade or so developing in situ transmission electron microscopy techniques that allow us to follow chemical transformations with near atomic scale resolution. And we can also introduce light into the transmission electron microscope, so that way we can see how light can drive out of equilibrium reactions. So if any of you make it to San Francisco, please don't be shy to reach out to me and come visit Stanford. I'd love to show you our in situ environmental TEM with optical coupling capabilities. Some other work that my lab has conducted is really developing new optical materials that are sensitive to mechanical forces. And I know we had the organizer here earlier, Youngjun, of the Upconversion Gordon Conference, which is happening this June in, I believe, the East Coast, New Hampshire. What we've done is develop nanoparticles that absorb infrared light and emit visible light, and the color they emit changes depending upon the applied force on the nanoparticle. And then we've been able to deploy these both in organisms, here, for example, in C. elegans worms, to be able to use the color readout to quantify the amount of force the worm is pressing on the nanoparticle or exerting on the nanoparticle as it chews and digests and excretes its food. So we can now start to correlate mechanical force with electrical signals in vivo. And we're really excited basically to understand kind of the basis of various diseases. And I wanted to mention this work because since we're at an ACS and KCS publication summit, I'm really grateful to ACS for the opportunity to have published many of these studies. I apologize that the resolution is not high enough to see it on the screen, but most of our materials development has been published in either Nanoletters or in JACS to create these upconverting nanoparticles. But like I said, today, kind of focusing on these post-pandemic transformations is focused on, bacteria. And bacteria are kind of the underdogs of the media now. I think for the past 3 years, viruses have kind of been the leaders of most media reports. But bacterial infections have a global impact. And if you look just to developing countries, about 30% of all deaths are due to a bacterial infection. And more than 10 million cases of tuberculosis, which is a bacterial infection, emerge each year. So tuberculosis is now an ongoing endemic. And by the year 2050, the World Health Organization predicts that antimicrobial resistance will be the leading cause of death, with over 10 million lives claimed each year. And that's because antibiotics are generally overprescribed due to the long culture times it takes to be able to detect bacteria and to determine their drug susceptibility. So to put that in context, if you wanted to detect bacteria, whether that's in a clinical sample or an environmental sample, you would generally collect the samples and then bring them to a central laboratory. And then you would introduce the samples usually into a culture vial, shown here in the center, and then put that into an incubator. And at the bottom of this culture vial is essentially kind of like a squishy gel or a squishy matrix that changes color as the bacteria divide and multiply. So we're able to detect the bacteria by looking essentially at changes in absorption in these culture vials. But then you have to identify what sort of bacteria it is by using either molecular methods or mass spectrometry. And then once you've identified the type, then you need to use the cultures to be able to do antibiotic susceptibility testing. So you'll generally put the bacteria, for example, on an agar plate, as you can see here in the image all the way on the right. And then you'll put down different antibiotic patches, and where you see the bacteria dying, then you know the bacteria are susceptible to that particular drug or that particular antibiotic. So you can see in this image there are 3 white antibiotic patches, and the bacteria are susceptible to all 3 of those antibiotics. But this process of culturing bacteria and then detecting the antibiotic susceptibility can take about 2 days, even in the most state-of-the-art laboratories, due in part to the long times to culture bacteria. It's just a naturally slow process. And with tuberculosis, it's a much more slow-growing bacteria, so it can actually take over 40 days to culture it, even though it only takes an instant to be able to determine if someone is TB positive, because it's such a long bacteria or such a long bug that you can identify it on a microscope slide, but you need to allow it to divide and multiply in order to be able to do the antibiotic susceptibility testing. And according to the CDC, about 50% of patients are either unnecessarily treated with antibiotics or are treated with the wrong type or dose. And that's because the consequences of not treating someone who might have a bacterial infection, who does have a bacterial infection, are so severe. Like, time really matters for many of these bacterial infections. So you want to treat someone in the event they do have a bacterial infection. But then if they don't, bacteria start evolving to be resistant to the antibiotics that were given to the patient who didn't need those antibiotics or who needed a different type of antibiotic. So we know that there are many advances now in synthetic biology and CRISPR, and bacteria can be engineered to have distinct colors. But we wanted to develop a platform that didn't require any synthetic biology or any engineering of particular species and strains. Instead, we wanted to rely on the natural color of bacteria. So what we decided to do starting about 5 years ago was see to what extent we could use Raman scattering as a label-free method of determining the bacterial species and strain and also its antibiotic susceptibility. So Raman scattering is inelastic photon scattering, and you can think about the bacteria as essentially kind of a bag of water that also has metabolites and lipids and proteins and glycolipids and glycoproteins kind of decorating the surface. So we send in a laser of a particular color, and then we're going to look at the light that is scattered to longer wavelengths, so the Stokes-shifted wavelengths of this Raman scattering. And that provides us with essentially a fingerprint of what the cell is. And it's going to be a relatively complex fingerprint because we have essentially the ensemble average of all of the molecular vibrations of what is in the bacteria. And when we look at single cell spectra spanning 31 bacterial species and strains that are most commonly seen at Stanford Hospital, we find that the Raman spectra kind of look similar, but also kind of look different. So there are some species that look quite differentiable from each other. For example, E. coli looks quite a bit different from Staph aureus. But E. coli looks quite similar to Pseudomonas pneumonia. It also looks quite similar to Group A strep. So we collected thousands of examples of these single-cell Raman spectra, and then what we did is try to train a convolutional neural net. So the idea is that depending upon how the bacteria land on the substrate, you might be interrogating kind of a different angle or a different portion of the Raman spectra. But if you use many of the algorithms that have been developed for machine learning of images and adapt that to spectra, you can essentially train the network to be able to identify a spectra it hasn't seen before if it's seen enough spectra of similar types. So we used basically a ResNet architecture, so a convolutional neural net, where we input the Raman spectra of many of the single cells that we already collected. And then once you've trained the network, you input a spectra that the neural net hasn't seen before. And what comes out is a series of predictive scores as to the probability of whether the spectra that was fed in belongs to a particular class of bacteria that it's seen before, or potentially to a class of bacteria that it hasn't seen before, in which case it can say, well, it seems most similar to this other bacteria. So if we look at just a subset of 6 bacteria, here's Staph aureus, Staph epidermis, MSSA, you can see that most of the predictions are actually accurate. So we know the ground truth from mass spectrometry, and then we can see from our single cell Raman spectra that took essentially a second to collect what the neural network is predicting. And you can see most of the hits are along the diagonal, so that means we have a correct prediction for the bacteria. There are some misclassifications, so those are the off-diagonal components. So you can see, for example, Staph aureus strain 3 is sometimes misclassified as this isogenic strain of MSSA. And we trained on 60,000 single-cell spectra and then tested it on over 12,400 spectra in this initial work. Now, if we zoom out to the full confusion matrix of those 31 species and strains that represent over 95% of all patient samples seen at Stanford Hospital, you can see that the average strain level accuracy is about 82.4%, which is not bad given that we had trained on a relatively small amount of spectra. But if we group the bacteria according to the first choice antibiotic that a clinician might use, you can see those groupings here in the different colors. And you'll see that most misclassifications were within that same treatment grouping. So the average first choice antibiotic treatment accuracy is actually 97.1%, which is much closer to what mass spectrometry would give. Now, those were on cell line samples, basically dispersed on a dried substrate. We wanted to know to what extent could this translate over into clinical samples. So what we did is take 30 patients, again from Stanford Hospital, where we knew the ground truth from mass spec as to what bacterial infection was. And then we took our cell line samples as basically the trained data and then fed in the clinical spectra that we obtained from these 6 patients who had confirmed positive E. coli infections or MRSA infections and others. And you can see that for the most part, the neural network does a pretty decent job, although there are some suboptimal predictions, meaning that the network didn't predict the right first choice empiric antibiotic, but maybe the second choice. But then once we could use that clinical data and feed that into our neural network, when we saw an additional 30 patients, we now got much higher classification accuracy for the first choice antibiotic treatment. So over 99% accurate. And interestingly, we don't need to have many cells to get accurate prediction. So we could look at roughly 1 to 10 cells, meaning that you don't need to have a large concentration in cells, and kind of hinting at the ability to be able to do culture-free detection of bacteria. So in that particular work, we had hints that we'd be able to detect the antibiotic susceptibility, but we were really only looking at the empiric first choice antibiotic. We weren't correlating our results with the known minimum inhibitory concentration and the antibiotic susceptibility or resistance profile. So what we did is try to see if we could determine what antibiotic the drug would respond to and which antibiotic it would be resistant to. And we did this on tuberculosis. So this is a huge endemic. In the year 2020 alone, 1.5 million individuals died from tuberculosis and 10 million individuals contracted the disease. So what we did is look at tuberculosis that we knew was resistant to 4 of the frontline antibiotics: amikacin, moxiflacin, isoniazid, and rifampicin. And then we also had the wild type tuberculosis that is susceptible to all of those antibiotics. And then what we did is collect thousands of examples of single cell spectra where we knew what the resistance profile was. And by eye, it's very difficult to see what the differences are between those tuberculosis spectra. But then when you project the spectra onto a two-dimensional plane, for example, using a T-SNE or a UMAP, here each dot corresponds to essentially a dimensionality reduction of that spectra. And you can see there's actually pretty good clustering between the tuberculosis that's susceptible to all antibiotics and the tuberculosis that's resistant to either amikacin, isoniazid, rifampicin, or moxiflacin. And we have over 98% predictive accuracy when we look at these resistant strains. Now, instead of just feeding the spectra into a neural network, we wanted to know what are the features that it's latching onto. And you can use different models like autoencoder models to try to understand, like, is it the peak intensity, the full width half max, the gradient? In this particular case, what we wanted to do is provide interpretability to the Raman spectra. So what we did is essentially remove peaks from the Raman spectra and then rerun the neural network and see how the classification accuracy changed when certain wave numbers were removed. So you can see that the accuracy drops down from its baseline accuracy over 95% down to below 80% just in certain bands. So when the accuracy drops down, you know those bands are really important for feature recognition or classification. So with tuberculosis, it's known that as the bug becomes resistant to certain drugs, basically it's growing a thicker mycolic acid wall. And here we have vibrational bonds that correspond to the CC stretch of mycolic acid, as well as a CH2 twist and a deformation thereof, and also a C double bond O stretch. So we only have a few bands that are really important for high accuracy classification. And those bands in this particular case correspond to known pathways or resistance mechanisms. So en route to clinical translation, we wanted to be able to look at different genetic variants of tuberculosis. So here's just an example of two drug-resistant TB, the amikacin-resistant strain and the isoniazid strain. But we have 3 to 4 genetic mutations that are just single point mutations in one gene fragment. And we want to know to what extent can we determine not only the resistance profile, but also which genetic mutation gave rise to that resistance profile. So first of all, encouragingly, you can see regardless of the genetic mutation, we can still clearly distinguish between different antibiotic resistant bacteria. But then even when you're looking kind of within a distinct antibiotic resistant grouping, we have close to 70% predictive accuracy for which particular genetic mutation there might be. So for example, in isoniazid, we're looking at the CAT-G gene and there's just one mutation, for example, a C to T mutation or a G to A mutation. So I showed you earlier how we have this wave number interpretability. And what you can do is start reducing the number of wave numbers that the neural network is using and see how the accuracy of classification drops down. And what we saw is that the classification accuracy actually remains quite high, even when you just have a few hundred wave number features. So that actually allows us now to create a miniaturized and low cost Raman spectrometer. It costs just about $1,000. And here's my student Baba holding it. So by knowing which wave numbers are important for classification accuracy, we can minimize the hardware constraints for deployment in the global south. And here using the handheld Raman spectrometer and looking at different sputum samples with spike-ins of tuberculosis, we're able to identify the drug susceptibility with very high accuracy. Okay, so far I've shown you mostly dried samples or sample in sputum, but what if you want to be able to detect bacteria, for example, in blood or in wastewater? You need to be able to handle these more complex liquid samples. So my student Loza, who's now a professor at MIT, started to look at liquid well Raman spectroscopy to see if a lot of the principles that we had seen on dried samples kind of held over to these liquid wells. And once you're in liquid, even though Raman spectroscopy still gives you pretty good signal, it's actually nice to have nano antennas that enhance the signal, especially when you have a lot of masking of your signal, for example, from red blood cells or from wastewater. So we looked at gold nanorods of different radii or different aspect ratios. And those have different resonances compared to the excitation wavelength. And for the most part, we're working at a wavelength of 785 nanometers. And what we found is regardless of the nanorod aspect ratio, we're still getting pretty strong enhancements. And as you might expect, it does depend a bit upon where your resonant wavelength is for your gold nanorod with respect to your pump wavelength and where you expect the Stokes shifted wavelength to be. But interestingly, we kind of would have expected this red or like this longest wavelength resonance to be the strongest enhancing nanorods. And it turns out it wasn't always the case. And that's because you need to think not only about the optical properties of the nanorods, but also the charge of the nanorod. So bacteria tend to be slightly negatively charged. The nanorods are positively charged. So you have to also think about the electrostatic interactions in liquid between the bacteria and gold. And staph epi tends to be one of the more negatively charged bacteria. So by looking at cryo-electron micrographs of the bacteria in water, you can see that gold nanorods tend to cluster around the staph epi. E. coli is one of the most positively charged bacteria, and you can see the nanorods are kind of around the E. coli, but not as closely clustered around the bacteria as with staph epi. So if you have just a few bacteria and a lot of other cells that are in your liquid, what we wanted to do is essentially digitize the sample. So kind of think about printing it into tiny little single cell droplets. And in that way, then we could collect images now that we know which wave numbers correspond to which bacteria to be able to say like, oh, that droplet has a bacteria in it, or that droplet has just red blood cells in it. So my student Fariha Safir and I collaborated with Pierre Kuryakov. He's one of the pioneers of the original inkjet printers. And what we did is essentially adopt kind of an inkjet printer, but to have an open well plate with a piezoelectric transducer beneath it. And then what we could do is put complex samples in essentially a well. And then depending upon the frequency that was applied to the piezoelectric transducer, print tiny droplets that contain just a few cells. So we could print milliliters of liquid within just a few seconds. We're essentially printing at kilohertz rates. And here you can see the droplet being ejected by using stroboscopic imaging. And like I said, depending upon the frequency that we use on the piezoelectric transducer, we can tune the droplet diameter that gets ejected. If we want to print just a few cells within each droplet, then we're going to want to tune the frequency to be as high as possible. So that way our droplets are just a few microns across. And here you can see the printout of the Stanford logo just with fluorophores in each droplet. So we can process milliliters of liquid just in seconds. And the interesting thing is that the bacteria survive the printing process. So if we print on an agar gel, you can see right away, it's almost impossible to see each of the droplets. But then as you wait over time, you can see the bacteria still continue to grow and multiply. So that's great, especially if you have a sample that has only a few colony forming units or a few bacteria per milliliter. You still want to know that you have the chance to find them if you needed to be able to culture them. So we printed the bacteria along with gold nanorods. And when we optimized the printing to be stable for these very different size components, we found that we didn't get any coffee ringing of the nanoparticles. Basically, the nanoparticles were still electrostatically bound to the bacteria. And here you can see a nice zoom in of those staph epi with the gold nanorods around it. And then what we can do is take scanning electron micrographs of each individual droplet so we know exactly what's in the droplet, but then we also can do Raman spectroscopy of each droplet. And we see, first of all, that, for example, with staph epi or E. coli or red blood cells, that when we have the nanorods, we get strong signal enhancement. It's very challenging to be able to see the bacterial signal or the cell signal without any nanorods. So we get strong enhancement. And then by training a neural net on basically what droplet Raman spectra look like based on kind of the number of cells that are in each droplet and the type of cell that's in each droplet, we can clearly identify distinctions between, for example, staph epi, E. coli, and red blood cells. We can also do this on mixed samples where we have combinations of red blood cells and bacteria and nanorods. And we can have either an individual type of bacteria or a mix of bacteria. And then again, what we can do is we know the ground truth from both mass spectrometry and also scanning electron microscopy, but then we can collect the Raman spectra, again, train our neural networking, we can get over 87% accuracy in terms of identifying which constituents are in which droplet. And the neat thing is that we don't need to have a large number of bacteria in any given droplet. In fact, you can just have one bacteria in a droplet and a large number of red blood cells. And here that's represented by basically the ratio of staph epi to red blood cells being basically less than 1. And even with a very small amount of bacteria in each droplet, we can still identify that there is a bacteria in the printed out sample. So essentially we can just scan over the sample and say, well, we know from the Raman spectra there must be bacteria present. So we wanted to know if we could adapt this technique from blood to wastewater. And we've seen that throughout the pandemic, wastewater-based epidemiology has been a really powerful way to follow the evolution of viral infections and the presence of a virus in the environment. But if you wanted to scale this to bacteria, it actually becomes very challenging because not only are there a large number of species and strains of bacteria, but also if you wanted to do enzymatic-based amplification or PCR, often in wastewater, there'd be too many salts and other kind of materials that would inhibit the amplification process of the enzyme. So the idea was to use our insights basically from liquid well Raman. So we took wastewater basically from Stanford, just passed it through a simple filter and then spiked in known concentrations of bacteria. And again, you can see even in wastewater, when there are a lot of charged salts, when you have the right nanorods, they still want to bind basically to the bacteria, regardless of what the bacteria are. And what we could see is that, as you'd expect, compared to just water or formalin, basically overall the Raman signature goes down when you have wastewater because there are a lot of other peaks to contend with. But when you have the nanorods, basically the nanorod concentration gives rise to an enhancement of the peaks that we know to be relevant for E. coli or staph epi, for example, 1002 or 1315 or 1441. So what we did is work with a nanorod concentration that was somewhere in the sweet spot of right around 60 micrograms per mil. And then what we did is vary the concentration of bacteria that were spiked into the wastewater samples, all the way down to 100 colony forming units per milliliter. And what we found is that by scanning the laser over the wastewater, we could still detect even down to 100 colony forming units per milliliter in wastewater. And we could also identify with over 85% accuracy in some cases, basically which bug was in the wastewater. So Staph aureus right now is the one where we have a little bit more work to do to get it over 80%. But I think it's pretty encouraging that with extremely small levels of bacteria in wastewater, we can still detect that bacteria are there. And then we can also identify which type of bacteria it's there. So we're really excited to apply this to wastewater-based epidemiology of pathogens. In the final few minutes, I just wanted to share with you work that we're doing beyond metallic nanoparticles and basically using silicon nanostructures and CMOS compatible nanostructures to be able to do more sensitive molecular detection. For example, if you didn't want to just detect a cell, but if you wanted to detect metabolites or peptides. So what we did is design essentially a silicon on glass surface that allows for very highly efficient Raman scattering, as efficient as the plasmonic nanoparticles, but it's essentially a more uniform SIRS enhancement since we can control essentially where the enhancement is occurring. And there's also no sample heating. So especially if you want to do dynamic measurements, this is a pretty powerful method because you're not going to be heating up your molecule because we're working in a spectral regime where silicon is transparent. So here we have a very sharp spectral resonance, basically at the pump wavelength, and then a broader resonance where we expect the Stokes-Raman scattering. And what we do is pattern these kind of high quality factor or resonant silicon nanostructures over our entire chip. So here you can see how the electromagnetic field is enhanced over each structure. The quality factors are in the thousands to tens of thousands. And we can pattern about 3 million sensors per centimeter square just on a simple glass substrate using semiconducting fabrication methods. So since we know which features correspond to certain molecular vibrations or certain cell transitions, what we can do now is essentially either scan the detector plane or scan the incident laser. And now we don't need to do kind of confocal scanning of each spot. We can essentially do imaging-based Raman spectroscopy or imaging-based spectroscopy. So here you can see an array of about 200 resonators. and they're each lighting up at different frequencies based on which molecules have landed on which individual resonator. And from that, we can reconstruct the spectrum. So as my final scientific slide, I just want to share with you how we can use this platform to detect peptide monolayers. So here you can see Raman scattering spectra over an individual resonator. When you are off the resonator's peak wavelength or resonant response, there's essentially no Raman spectra that can be detected. But as you go on to the peak of that high quality factor resonance, now you can get strong Raman scattering signature, even when there's just 24 attograms of peptide that's in solution. And we wanted to see to what extent we could differentiate just small atomic differences in peptides. So here we looked at a 4 amino acid long peptide, RGDC, and compared that with RGDS. So swapping out the cysteine and the serine amino acid, which corresponds to just a single atom difference between the sulfonyl and hydroxyl group. And we can differentiate those really well. And we can also differentiate a 5 amino acid sequence, GRGDS. So with that, I'll summarize by saying I think there's a lot of power in merging advances in nanophotonics with advances in AI and machine learning, especially when it comes to rapid and accurate pathogen detection. So as a platform, I think plasmonic nanoparticles and high quality factor dielectric metasurfaces both have their own unique advantages when it comes to being able to do bacterial and molecular detection. In both cases, you can confine light to ultra small volumes. The gold nanoparticles have the advantage of being electrostatically attracted, basically to almost all bacteria, especially if you want to do liquid Raman. And then these metasurfaces enable more uniform SIRS enhancements without heating, so especially useful if you want to do molecular dynamics. And then we also developed this acoustic droplet ejection platform that allows us to do picoscale printing of complex samples, and basically to be able to process lots of liquid in a very fast amount of time. So essentially we can collect real-time Raman spectra as each droplet is being printed. And then I shared with you how Raman scattering and ML can enable pathogen identification as well as their drug susceptibility, including for a variety of pathogens seen at Stanford Hospital for bacterial bloodstream infections, also for sputum, and finally for wastewater samples. So with that, I'd like to thank all of my students for their incredible efforts and all of you for your attention. I'm happy to take any questions.
Thank you for your inspiring talks. I think that all of you have a lot of some questions, but we have a time limitation. So only just urgent one question. Yes. Yes. Yeah.
Camera angle. All right. I have two questions. Easy one first.
No, only one, please. One is later.
Yes, one.
Please. Yeah.
Hey, come on. So this MRSA, you have shown that there's one little discrepancy in the case of MRSA, this antibiotics resistant bacteria. It's because it is simply because it's antibiotics resistant bacteria, or any other reason why it has some just little of like a defective problem. That's my first one. Second one is the simple thing is a general thing that we have really good news that we can utilize the machine learning in analyzing many tens of or hundreds of Raman spectra, even complicated one. That's a good news. But at the same time, the bad news is traditional pattern recognition or chemometrics is now like almost like abandoned, but this machine learning. But as you know, as we have observed in the case of AlphaGo, this machine learning really heavily dependent on the algorithm, right? So depending on which algorithm you take by chance or by lucky way, the result can be drastically different, right? Please comment about that too. Okay.
Cool. I'll try to be super quick. So the MRSA, MSSA, that was initially on dried samples and clinical samples where we hadn't done kind of enough training in the clinic. I'd say from 2019 when we first published that to now, we've been able to get very high classification accuracy. And you can kind of infer that from the TB strains, where now we can even see a single gene difference. So it's essentially when you move to clinical samples, a matter of having enough variation, so that way you're not classifying based, for example, on age or blood type, but you can identify the drug susceptibility and the features that really matter for drug resistance or susceptibility. And like you said, the model that you're using can matter. And like if you want to start using some of these larger models, basically you can infer a lot more about the kind of molecular structure and which features are giving rise. So I think as we start training more and more Raman data, we're certainly not in a regime where you can use like some of the large language models, but the model matters to a certain extent in terms of accuracy, but it only gets better when you start using convolutional neural nets, and basically the more data you have, the more insight you can get into different biological pathways. And happy to chat during coffee. I know you said be quick.
Okay, thank you very much. Okay, we move the next speaker. Happy to introduce Professor Jinwoo Chun. He's the professor in Yonsei University, Department of Chemistry, and also the director of the Center for Nanomedicine Institute for the Basic Science. He also served as an associate editor in the accounts of the chemical research. Okay, let's warm the welcome the Professor Chun.
So, thank you so much. And because Young-Duk spent a few minutes on behalf of myself, so I'll make it short. So my talk is more into materials, but you could say that the materials can be pretty much well designed and then executed over past, I think, 2.5 decades. So, but I think these are something that people are really interested in. I mean, you can make any type of, any components, any shape, but still, I think we are still in the process of something that's in the metastable, kinetically trapped state. I think those are very important to explore. And then I think we have seen many beautiful talks about this afternoon. So I'm not going to go over there, but I think we found that some of these materials can be quite useful for biological studies, so, which is our main interest. And I'll tell you just one material system among many possible that we have explored over the years. So this is the question that I have been interested in over the years. And so here we have magnetism. Here are biological matters, including anything from cells to our human body and so on. So can you really see by using the magnetism biological system much clearer, right? Of course we do. I mean, MRI. I went Illinois for my graduate school, and then Andy Gors was my also committee member, and then there was a professor Paul Rothbard. He won Nobel Prize by inventing MRI and so on. So it has been well executed, and I've been work on developing contrast agents and so on. But the other thing is that if you can regard it as a reading, and how about the writing? Can you really use magnetism to either activate or deactivate the biological system? So that's, I think, a quite interesting question to explore. And one is, can we do it first? And second question, then how can you do it? Then why do we need nanoscience or nanochemistry in that regard? All right, so that was the question. Here, see the magneto, right? So this is from the movie X-Men phase, right? So the and he can remotely control metallic subject, and it's not something that we can really think of, right? Can we really do it, and what are the tricks that he's playing with? So, but two things. One is wireless, and then remotely control, and then the matter of the subject is metallic, or I mean, it's supposed to be magnetic in that regard, because that's why he's characterized as a magneto. So then when we see in nature, we could also see that something called the magneto reception. That means that somehow some biological creatures, they may sense geomagnetic fields, and then the geomagnetic field may guide them for desired position in some way, so serving as a magnetic GPS. And can it be true or not? So there are many hypotheses, but still, it's still under debate. But people have been very interested in this topic over, I think, the past few decades, and still we don't know much. So now we have utilized optical system very intensely, and then we have developed just heard many beautiful talk, including Jennifer's. But light is wonderful and probably the most important invention in our current modern science. But still, when it comes to the biological system, it has a huge limitation, especially the live animal, right? So maybe it works for the cells and genes and so on. So the penetration issue is huge. So that means that light basically interfere with the biological system, protein, genes, and so on. So it doesn't penetrate deep enough. So what we have been interested in is, what if we invent or discover something that can emulate the optical system? For example, we have a flat, very well-known system for optical system. Can we have a magnetic system? And somehow it is operable, but with a magnetism, somehow, something like that. So we call it MRED. We have developed a system that can be utilized in that concept and storm instead. And then how about high resolution MRI imaging? And the third one is probably you are aware of that people have been very interested in using optics to control the neuronal system. So that's called the optogenetics. And then can we have a magnetic system in that regard? So the third one will be my talk of discussion today. So here, the simple system, because it is so simple, that all you have to have is just triggering the action potential by using the mechanical force, but the mechanical force is needed to open some ion channel. It's called a mechanosensitive ion channel in neuron. And then once it is being triggered by mechanically open, and then calcium goes in, for example, cation goes in, and then it generates the action potential, and then it relay into the other neurons, and basically it is activating the neuronal system. So here what you need is a few piconewton of force, or a few need, probably 3, 4, 5, and so on. So then how do you do that? So here is a landscape of force scale. As you can see here, start from micron, nano, to pico, to femtonewton. But you only need a few piconewton, nothing else. If it is too weak, it's not going to do anything. If it is too strong, it will destroy the whole system. So that's how I think the ion channels or proteins have been developed over the years, right? Many, hundreds of millions of years. So all you need is the force that can be regulated by, in that region, only few piconewton of force, right? So people have thought about this for many, many years. And then this is one good paper that came out in 2016. So the idea is very simple. So what if you introduce ferritin as a genetically engineered, so that it generates ferritin. Ferritin tend to collect iron, right? And then they mineralize the iron to form iron oxide in situ in a live animal system. Then what you can do is you use a magnetic tweezer to pull it out from the membrane, then there generate tension, and it will basically open the ion channel. That's simple. So they tried, and what happened was that people tried to reproduce the study, and then they couldn't, actually, they couldn't. And then 3 papers came out, in 2019. I think this is one of them. Say it is not working. It's not working. So I mean, then why? And then they came into the discussion saying that basically the ferritin, it stores iron and mineralize and so on, but it makes fatty hydride. Typically that's anti-ferromagnetic. That means that the spins are anti-parallel, and then all the remaining ones are on the paramagnetic, small spins on the surface of a nanoparticle. So if you do back of the envelope simple calculation, the pulling force is below attonewton. And then when you come into the room temperature thermal fluctuation, is more than 10,000 times smaller than thermal energy. That means magnetic spin is lost. It's not magnetic at all, such that you cannot really pull the ion channel. The membrane is not going to perturb at all. So there are some biophysicists, they just say, oh, I mean, biologists, you should know physics 101. Maybe not in high school physics and something like that. So there is a big discussion about that. So, so all you need is a force, right? So the force, I mean, it could, different from force is, Jedi force. The force, generation of force is not simple error in biology because it's very selective force, such that somehow you can do something. Okay, so let me just. So what our thought was that 2 things. One is the force has to be generated in far away. That means that it has to be long working distance. I think that's very important. The other one is the force has to be large enough. So, but we all know that we have a magnetic compass, right? There is a north and south, and then it will turn, right? The compass, depending on the position of the compass, it will rotate toward the north and south. By doing so, you can generate, torque, right? The torque is there, but somehow it's a very long distance, force generation. So what we have done is that somehow you have to have a proper, well-designed magnetic material. We call it M-torquer, magnetic torquer. And then the second thing is that, how are you going to test it into biological system? So you really need a bona fide mechanosensitive ion channel. But mechanosensitive ion channel has not been well studied until very recently. Very recently, that means that about 10 years ago. Before then, people thought that, oh, here is a mechanosensitive ion channel, but still, it wasn't quite well established. And then third thing is that, how are you going to generate torque? But probably probably better than me, you can also know the answer. So the first thing is that you have a magnetic nanoparticle which can generate large susceptibility. That means that a single nanoparticle cannot do it. So we synthesize nanoparticle, there's the octahedral nanoparticle, to generate this a little bit of enhanced anisotropy. That means a small residual magnetic coercivity. And then the simple way of enhancing is just collecting into a one spot. That means they're just coating into 100 nanometer sphere of polycyan or silica. And as you can see that the coercivity doesn't really enhance, but only the magnetic moment. So that could be quite effective for serving as the torquer. So, and then next thing is that how do you create torque? Okay, so when you have two magnets, okay, so you have a north and south. The gradient field goes on very quickly. So the working distance is only a few millimeters in general. So you have to really stick into the object very closely. And now you want to have a long distance torque generation. As you pull it, further and further, the field goes on, and then there is nothing in between. So let's say you start with 8 centimeter, you have a magnetic field, but if you go down to 16 centimeter, there is nothing in there. So, but people have already, developed this concept. You put more magnets simply. So let's say you had 2, top and bottom, it will generate, different type of field line. But if you have 6 of them, for example, you can have a more like a north and south field line. And then the diameter of this system has been increased twice. That means that you can somehow modulate the working distance of the system. And then next thing is that you rotate the whole magnetic system. So we call it circular magnetic array. And then accordingly, the particle will rotate. So that's simple. And this is the system that we have developed over the years. So you have a north and south here. Okay, let me just, okay, this is better. So you have a, subject of your interest, north and south. So, and here is a membrane, cell membrane. Here is a nanoparticle. And nanoparticles tend to align into the magnetic field, right? And then the next stage, you rotate this whole magnetic system, so that like a circular magnetic array. And then you can have a choice of a duration, and then the angular velocity. We all know that. So then what you are doing is that you can generate magnetic pulsation. And then if you have a certain frequency of a rotation of a CMA, then basically you are turning on and off of the gating process. So this is a direct force generation, and it's a bio-orthogonal. That means that there's no leak signal error. And then the kinetics can be controlled easily. So this is the system that you see. So this is a CMA, and then the diameter is about 16 centimeters. And then here you see the 6 of them. And then this is only when there is a magnetic field, the pulsation is on. The calcium influx is, as you can see here, the light, the red light is just showing that the fluorescence is on. That means that this is a calcium reporter, so the signal is on. So this is fairly fast and reversible. Then we need some ion channel, but it has to be mechanosensitive. And then this one actually was discovered by Adam Potapotian at Scripps. And then this one was developed, published in 2010, not too long ago. So it took probably just over 10 years to win the Nobel Prize in physiology and medicine. And because this is so elusive, mechanosensitive ion channel, it has been so difficult to study. But finally, and now there are a series of mechanosensitive ion channel discovered by people, and then they call it piezo, means pressure, right? So it's a biological system, and it's all related to our, balance, weight, and sensing, and then so on. But something that we are not really aware of. So that many people are calling this related sixth sense. It's hidden to our system. Without this, we cannot survive. It's that important, but it's so difficult to study this ion channel. I utilized that system, so I made a visit to Scripps and met with him in 2014. So it took almost 9 years to get here. So I talked with him, we discussed. So I got the gene from him, the DNA, and we put it into the virus. So here you have a neuron. Neuron doesn't have any mechanosensitive ion channel. So you have to genetically engineer the neuron so that so you inject the virus will deliver into the neuron, and the neuron will express mechanosensitive ion channel. This is a piezo, and then as you can see, the piezo is this with over the neuron cell, and then we introduce the nanoparticle. Okay, so nanoparticle that I mentioned to you has a conjugate with an antibody, and antibody will bind into the piezo1. Piezo1 has some tag, genetically encoded. It's a lot of genetic engineering. You need that. So the particle will only bind into piezo1. Okay, and then you apply the magnetic pulsation, and then it will of course open the ion channel and so on. So we tested this into a mouse. Okay, so and then we are interested in whether this is really working or not, and then so we injected the motor cortex M2 region is related to the motion of your legs, arms, and so on. So the red signal indicates the calcium influx is indeed happening, and then one behavior study is more important in neuroscience. So here you see a mouse just sitting there. It's not moving, but it's there. You can see that it's breathing, and because it's nocturnal, right? They don't like mobile that much unless you see a food, right? So they just stay there. But once you start the magnetic pulsation, then you see huge oops. Somehow. Oh yeah, right. Okay. You see that? So this is the magnetic field off, and this is a mouse injected in the M2 region in the right hemisphere. And then when the M2 region is agitated, usually the left pole is moving, more actively moving, such that you are rotating this way, counterclockwise. And if you do the left, and then it's that. So we were able to see that indeed magnetism can activate neurons through the mechanical stimulation. Mechanical stimulation, you choose the force range of a few piconewton, and indeed it's working beautifully. So now then what can you do, right? So you can do a lot. I mean, you have to first show that this is activation of neuron. You have to somehow show the deactivation. I think that's very important. And the other one is cell specificity. That means that you have a, thank you, 100 millions of neurons. That's what and then how are you gonna target those neurons? I think that's a big issue. And then later people are, and this is more interested in neural circuits. Neural circuit is very complicated, and how are you going to do that? Now, people are using the optical system, but still, you cannot really go to penetrate through the skull and so on. So you could do a lot with a remote system rather than the hooked optically wired system. That's a huge difference. It's like you have a cell phone versus you have a wired old type, the telephone, right? So handset. So you could do a lot of biomedical tests. Here you have a food. This is a related eating system. So this mouse is not it's just moving around, but somehow you can induce the enhanced consumption of food, right? And then because you have to, there are some neural type that you have to agitate, but you can make it as normal as possible. But this pulsation somehow simulate the enhanced fooding. But this is all it's not just random. So those cells on specific neurons are being triggered in certain neural circuit system. It's not just random neurons. But through magnetism, it's possible to do so. And then you could have a reverse. That means that sometimes you are very hungry, but you make you less hungry, or you lose just appetite. That's also possible. And then this one is related to Parkinson's disease. You can also alleviate the Parkinson's symptoms, and then there are some control system that you can kill all the the dopamine releasing neurons, but somehow you can also enhance the treatment of Parkinson's disease. For example, these are 3 mice, but these 2 have damaged neurons. But this one has the magnetic stimulation over the 2-week period, 30 minutes per day, and so on. And then it almost recovered their damaged neurons and so on. So I think that's still ongoing project. So here is the summary of my talk. So people have been utilizing electric field, probably the electrodes to simulate deep brain and so on. And then the optogenetics has been beautiful. I think they have been like really in the shining new light into the system in neuroscience. So that has been really good. But there are also limitations associated with the optical system. So I think this is something that we are very interested to push in this field. And then it's more like this nanomaterial is very important. So this is my take-home message. So you could do a lot as a chemist to the development of not only the biological sensing, pathogen detection, and then even into live system, because neuroscience is probably the most complicated system you can look up and explore. But I think it's possible with a high precision with a nanoscience. I think that's the end of my talk. And then nanomaterial, we are trying to understand what's happening between nanomaterials and the biological system. I think we have some understanding, and then there are more to explore. Right, and that's it. And then thank you for your attention, and I should thank my funding agency.
Thank you for the very insightful talks. So we have already time is over, but just one quick question if you have.
All right.
Okay, oh, sorry. Oh.
Very interesting talk, Junimo. And I'm totally outside the field, of course. I'm just wondering whether heat may play some effect when you apply to magnetic field and you have these particles inside here. Whether it's only like a magnetic field induced externally, or you also have some heating.
No,
of the
we are right. I
particles?
think that's very good question. But the heat generation is through either Néel relaxation or Brownian relaxation. That means that you somehow excite the magnetic spins and then let them relax. But this one, the rotational speed of CMA is 1 hertz, 0.5 hertz. What if you want to really agitate magnetic nanoparticle? It has to be 100,000. 500,000 hertz. So there is no heating error. So this is a system that's totally free of heating. And then all you have to do is just generate rotation. Rotation will generate Brownian motion. It will create heat, but the speed of rotation is, let's say, 1 hertz. It doesn't do anything. So there is almost no heating error. So this is 9999 whatever. Maybe there is femto very little, but we could say the heating effect is 0. So we have proven that, but theoretically it's impossible to have a heating in this system because some people are utilizing the alternating magnetic field, right? The alternating means almost a megahertz, but here is 1 hertz. So.
Okay, thank you, Professor Chen again.
Thank you.
Hello, my name is Yu-Sung Jung. I will be chairing the last session of this KCS ACS symposium. We have 2 speakers, and looking at the titles of the talks, the theme of the session seem to be chemical interactions and weak interactions for molecules and materials. We have one theory talk and one experimental talk. So to begin, we have Professor Sing Su from Fudan University in Chemistry Department. He holds a chair professorship in Fudan. And also he has been in an editorial board of various journals, but currently he serves as a founding associate editor of GX Gold. Floor is yours, Jun.
So thank you, Joe. So first of all, I would like to take this opportunity to express my sincere thanks to KCS for this kind of invitation. In fact, this is my first conference visit overseas post pandemic, and so I'm very happy to be here. And also, I noticed that my talk is a theoretical talk, and so I'm working on density functional theory. So I hope that from my talk I can give you a feeling what the status we are and what the challenges we are facing. So this is an outline of today's talk. And so, of course, the introduction part, I will show the functionals for the molecular application. Needless to say that density functional theory has made a tremendous contribution to the field. And so more than 90% of the theoretical calculations are carried out by using density functional theory. So according to the statistics, and people would say that this functional BSLIP usually run by using Gaussian. It's most popular for chemistry, but this functional PBE often run by VASP as most popular functional in material science. So when people talking about chemistry, we are focusing more on making bonds and breaking bonds. And when people talking about the materials, and so we are mostly interested in the bulk properties like band gap or something like that. So this is a more recent survey done by DOE in US, and to see what is a heavy load in their supercomputer centers. And you can see that more than 1/3 of the computational resources have been used on density functional theory calculations, and actually most of them are done with WASP. So my first question is here is so for some of the fields like catalysis, that's where small molecules meet bulk solids. So you need a functional which is both good for chemical bonding, breaking and forming, and also need a good description of the bulk. And in fact, so any of the gas phase reaction and the corresponding surface reaction, they are related through this Born-Haber circle. So maybe the computational catalysis will be less meaningful if the gas phase reaction, the small molecule, are not suitably described in the first place. So let me show you. So then please not disappointed by the DFT calculations, but I'm trying to show you this is a current status, and we should be aware of, and what we can do to face this challenge. So. For example, if we're interested in the CO bond dissociation, and this is a WASP calculation with PBE on the gas phase. And so experimental values like this, and this calculation value is like this, and so this bonding is too strong by 20 kilocalories per mole. And then when you do catalysis, you need this gas phase reaction. So if we're assuming the surface reaction are good, and then you will have to rely on the error cancellation. So no matter this is a nickel or rhodium, or no matter this is a 111 surface or 100 surface. And so for the catalysis, it's a complicated reaction network. So for example, here now we have the hydrogenation on the CO. And then for this reaction, PBE at WASP will underestimate the heats by 12 kilocalorie. So they are of the opposite side. But then again, you would expect the gas phase reaction error will be canceled out by all different kinds of matter, and then so you can have a good description on the surface. So if this is quantitative errors, and we can also have qualitative errors. So for example, for CO adsorption on the platinum 111 surface, and the experimental this will say this is on top adsorption. But all the calculation will predict this is a adsorption on the bridge side or the hollow side. So you can see this is because, so all the calculations will always stabilize the 2 pi star orbitals. So there are too many 2 pi back donations, less sigma donations. And so if this is favorable, then we may have the on top adsorption, but then you can see that this is a homonym of the CO and the band gap, and all the current DFT will not give a very satisfactory prediction on the orbital energy. And of course, we need a solution, and this is what we have been working on over 10 years, and you can see the homonuma and the band gap is in good shape now. So, so there are many functionals over these years. This is produced views from the Hartree-Wood to the chemical accuracy. And PBE is actually a LAN 2 functional, and BSRIP is a LAN 4 functional. And the functional we have been developing is fifth LAN functional. And so you get, this classification depends on how these functionals are developed and how, what kind of the descriptors or ingredients are used to construct these functionals. And so briefly, the new functional, we call it double hybrid, is because we're using Hartree-Fock exchange here, which is an initial position, and also the Gaussian of second order perturbation, which is initial slope, because both initial position and initial slope are important, and so we do this this kind of a double hybridization with a formal density functional theory. So of course, if we include this double excitation, and then the scaling is coming up, so we can do small molecules, but not the huge molecules. But of course, we are most interested in making chemical bonds. And then you would say that if only these 2 electrons are of opposite spin, and then this is important, do explicit calculation only on this part and leave all, say, parallel spin to be described by DFT. And in this way, you can reduce the scaling. And of course, only when the 2 opposite spin electrons are in close proximity, and they make a chemical bond. And this is most important. And this is not so important. So we left this be described by the DFT. And actually, this is a collaborate with Yu Sung Jun and also Bill Goddard. And so you can see the performance. So here is a general performance, and so with a common DFT, this is PBE and BSRIP for the heats of reaction. And so you can see that the PBE is actually not so good, and BSRIP improvement, but it's up to the functional we call XYG3 or XYGOS, and so correctly we call XDH functional, and so we can have a good description of the heat of a reaction. And of course, barriers are also important. And so you can see here, we have a heavy atom transfer, nucleophilic substitution, and also unimolecules and hydrogen transfer. And all of them, we can have a uniform accuracy for this kind of very high description. And of course, I would say this is also a very important message, because sometimes we have been working hard to do all different kinds of reaction mechanism, and we believe we have done a systematic reaction study. But actually, the functional you've been choosing will have bias on the mechanism, and so probably it's your functional that choose the mechanism rather than the true physics. And also for the non-bonded interaction, we have hydrogen bonding, charge transfer, dipole interaction, weak interaction, and peptide stacking. And you can see that it's also important to have a balanced description of all different kinds of this system. And of course, this is for geometry, and for bond distance, we have been testing on the covalent bonding, non-bonding interaction, and transition state. And so this is important to give a reliable description of the whole potential energy surface. So for over 10 years, we have been developing, doing methodology development, and now we have first order analytic derivatives, so we can do geometry optimization. We also can have orbital energies. We take the total energy derivative with respect to the orbital occupation, and so we can we can have this orbital energy. We can also have second order analytic derivatives, so we can have frequency and the NMR shedding constant, and so this shows the performance as our functional against the CCS DT, which is a very expensive method, and you can see that the agreement is very good for the NMR shedding constant. This normal DFT performance. You can see the error are huge, and now we can reduce this error. And so for the carbon, the 13 NMR, we can actually push the limit to 1 ppm. So if we have a good description for the molecule, and the question is, can we also extend this functional to describe the solids? So, so actually, there are many representative. So here are some representative MP2 implementation with periodic bound condition. So we are mostly focused on the FHI aims. And so for the canonical second order perturbation theory, then not only the electron integrals are important, we also have the new dimensionality problem with respect to the K-share size. And you can see for the normal DFT, the K grid convergence is very fast, but then if you come to the PD2 or XYG3, and then the convergence is very slow. Also, for the base set convergence, and this is also not as fast as normal DFT methods, but you can see that it's good to pay this kind of the cost in terms of accuracy, and this is for solid cohesive energy for a set of semiconductors, or so you can see that the cohesive energy for the new functional is very good, and so then we put these two together. One is molecular bonding and the other is cohesive energy for the extended system. And you can see that BSLIP is not good for the solids, but much better for the small molecules. And this is PPE's performance. This PPE is not good for the small molecules, but it's much better for the cohesive energy of the solids. And it's only up to the new functional that we can have good description of both. And so here I show relative stability of rutile and anatize. And so even with PPE, this will give wrong prediction of the relative stability, saying that anatize is more stable than rutile. And with the new functional, we can get the correct prediction. And this is for CO adsorption on the sodium chloride surface, and you can see that with scan, the adsorption energy is good, but the adsorption geometry is not so good. With this PPED3, the geometry is good, but then the adsorption energy is overestimated. This is also our new functional. But then I have to admit it, these calculations are very expensive. Can we gain accuracy and efficiency at the same time? And here you can see that the scaling, if you want to push the quality of this calculation, but then it getting much more expensive nowadays. So even MP2 at the periodic bond condition is only practical for medium-sized crystal system. So of course we know the solution may be. So for example, we can do this QMM embedding. So in Gaussian, this is called onion 2 scheme. So if this is our materials, but this is too big, so we can only treat it with a low level. But then we have an active sender, we can cut it out and then do a high-level calculation. We can combine these two, and so we can make this kind of hybrid model. Can we apply this scheme, which is often used in molecular science, to solids? So the idea is here. So can we do a WASP calculation with the PVC? And then can we cut out a class model to update this to a high level? So this is what we call that high level at XOPBC. We extend the onion model to periodic bound condition. And so you can see that we're starting from PBE at PBC, and then we do class model calculations using high level minus PBE. So of course, when this cluster is infinite size, and then this is a solid. But what we are really interested is this difference. So when the cluster size come to some number of n, and then we already reach this convergency, and this is a local nature of this correlation. So we can try this with molecular crystal. So say this is ammonia crystal, and then we can do the PBE WASP calculation, and then we can do the dimer calculation, trimer calculation, tetramer calculation with the high level, and then we can replace this differences, and so we can have very good results. And in fact, this is working. And so you can see that using this 23 set for crystal, for this XOPBC, you can always get the most satisfactory results for all the methods published in the literature. Then coming back to our surface reaction, and so here we show many of these examples. So this is for hydrogenation reactions, and you can see so for gas phase reaction, and PBE could be way off, and the new functional works satisfactory, and this is for CO bond dissociation, and this is for carbon-carbon coupling. But then the problem, the question here is, can we also describe this adsorption bond satisfactory? And so then what we are doing is we do a normal PBE calculation with VASPA. And then we cut out a cluster to do this double hybrid calculations. And so this is we show an example for the CO2RR on the copper surface. And with this, and the copper cluster could be like around 30. And in fact, for this kind of cluster calculation with the modern computer, it's only take additional 30 minutes on each structure. And so this is very, very efficient. And so to show, because CO2R, there are so many applications on CO2R, and compare recently do some measurements saying that if this is a CO2R, then hydrogenation of this CO2 is very important. And then he showed that for the PBE and the PBE plus D, there's a huge error on this adsorption energy, even up to 0.7 electron volt. And then you have so many DFT study on this CO2. How could this be possible? And so you can see that the error bar here for different benchmark set, the XYG3 calculations are very satisfactory. So we can directly using this computational hydrogen electron method to do this electro CO2RR on to CO, and using on top of the copper 111 surface and the copper 100 surface. And this is our calculation. And so this is as compared to the experiment onset potential on copper 111, the experimental value is 0.6. So this is our value. So on copper 100, and this is a 0.3, and this is our value. So this is a very satisfactory. And also for the experimental equilibrium potential, this is 0.1 electron volt as compared to our number. But then if you look at the literature number, everybody are using this. This is a PBE plus D, and this is a PBE. And you can see that the energetics are completely different. And even the rate determining steps are very different. So there are so many calculations nowadays on this system. So now we're also pushing this to the adsorption of small alkenes in zeolites. And with this kind of scheme, we do PBE calculation with the periodic bound condition, and then we cut out the class model to do the XYG3 calculation. You can see that the error bar is much more reduced also for this kind of system. So let me wrap it up. So, so of course, so I show you two ways that we can push the XDH functional from molecule to the periodic bound condition for solids. So this one is expensive, but it's serve as a benchmark purpose. And this one is actually very efficient. And so we can do the PBE calculation as usual, but then we can do the class model calculation on the active center, so we can have a good, improve the quality on. So here I also caught something in the literature. One is reproducibility in the density functional theory calculation of solids. And so for molecular calculation, the students doing calculation in MATLAB should give identical results from the other lab. But this is not the case for periodic boundary condition. So you may be, as a professor, you may ask the students to check the reproducibility of their DFT calculations on the solids. The other thing is nowadays we've been using machine learning a lot. All the machine learning data are based on the GGA calculations. And so you could see that, we are now seeing a real convergence. What the experiments want is what the series can deliver. But there's also a warning message. The truth is some errors come with the theory itself, we may never able to correct them. So this is also kind of a warning message. So as I said, that I'm showing the current status of the popular DFT calculation, but I do not deny DFT has made tremendous contribution to the current field. So so only we should keep in mind there are pitfalls here, and so many thanks to the group members and the collaborators. So with that, I would like to thank you very much for your attention. So thank you.
Questions and comments? We have some time. Okay. If not, then I could ask one.
Yeah.
So one of the take-home messages of your talk today is that one needs to be very careful what function to use to model mature systems, because
Yes.
PB could have very large errors. But your method is accurate, but still not applicable for routine large scale screening purposes, right? Do you have any sort of recipes or when one needs to be more careful so that PB might be wrong?
Oh yeah. Actually, it's really difficult to say. And now for the couple surface, so in terms of the CO2 RR, we are quite confident that many of the results could be in error or in serious error. So the students are happy that we on this system we can do many calculations, because this is very important to show new mechanism, and we also are quite confident our methods could be used to system like zeolite or MORPH, or this kind of things for predicting the interaction, in particular when weak interaction is really, really important. And currently, the students are working hard on the methodology for the platinum surface. If we can really make this on platinum surface, and then we can generalize our methods to all different kinds of system. So currently, PBE is most popular, but we should be aware of the pitfalls. Yes, yes.
Okay. If there are no other questions, in the interest of time, let's thank the speaker one more time.
Thank you.
Thank you very much, Sin. Our next speaker is Professor Young-hee Lee from Seungkyunkwan University. He is a professor in physics and also Department of Energy Science. He is leading one of the IBS centers. And he will be talking to us about van der Waals layered materials from synthesis to applications for today.
Thank you, Chairman. So I slightly modified this title of the work. Instead of having this van der Waals layered material from synthesis to application, which is probably too wide, so I decided to focus on this particular title, working on this diluted magnetic semiconductors. In fact, we are talking about this magnetic impurity in semiconductors, and in this audience, I realize I'm the only physicist working on. So in this sense, I'm purely impurity. And but I think since we are talking about this material, we talk about this synthesis chemical vapor deposition. So in this sense, we are still correlated. So I think we are okay. So let me move on. So let me first mention our collaborator, Professor Locke, working as a team leader in our center, and Seokjun and Bomsop, now Duan, and also this Byung-uk and Langan working on this magnetic materials, and Youngmin is working on the TM analysis, and also Mingyu is working on this noise spectroscopy, which is very necessary to understand what's going on, to see our this particular systems. So if you look at this periodic table, of course we have many different van der Waals layered materials, two-dimensional. Of course, this most famous system carbon, which is graphene. And if you look at this phosphorus, of course, we have many different polymorphs, this black phosphorus and black, not only black, but also this red phosphorus, and we have a white phosphorus, and we are talking about violet phosphorus as well. Now, if you combine these two materials with boron, with nitrogen, of course, it's insulating this boron nitride, still a perfect, stable 2D layered material. Now we are talking about something further, combining these chalcogen atoms with these transition metals. Then in such a case, we have many different classes of materials of the transition metal like chalcogenides. And of course, I've been working on this crystal growth of the material. And briefly, I can mention some of the, we've been working on this like a meter scale graphene film, but it's polycrystalline. And then we started to move on this special substrate, is single crystal copper 111 film, and we can make this single crystal graphene. And we can further move on to this multi-layer graphene, which is highly stacking ordered. That's also especially using this particular silicon copper alloy in this case. We are also talking about this hexagonal boron nitride single crystal, and in this case, we can make this a single crystal, this HBN, on this gold, liquid gold, and we can go further with coherent this heterostructures, even graphene heterostructured with this HBN. And recently, we can also make this a single crystal still wafer scale using all these TMD materials, in particular using this atomic sawtooth gold surface. This is all substrate dependent. So the key is the substrate, how to choose it. This is the key, and I think one of the audience, Sumin, is working on this seriously on this different substrate to make this single crystal growth from a chemistry point of view. So, okay. Now, for given the 2D transition metal like chalcogenides, we have many different phases, starting with hexagonal 2H, which is a semiconducting, and move on to this octahedral and distorted octahedral. All these are metallic, 1T or 1T prime. If you go on multi-layer, we can have this also rhombic TD phase and rhombohedral 3R phase. And what is more interesting that all these different polymorphs, different phases, are all intermingled with different very interesting physics. As you mentioned, there are many interesting physics we are talking about from physics, also from a chemistry point of view. So I like to simply mention there's plenty of room in the 2D van der Waals layered materials, in particular these TMD materials. Now, what I like to emphasize some of the very important feature in talking about this magnetic semiconductor material. One is a spin-off coupling. Spin-off coupling is simply proportional to this atomic number. So if we have this heavy atomic number, then we can have very strong spinor coupling. For example, like molybdenum or tungsten. In such a case, that changes many from the physics phenomena. As I mentioned, spin Hall effect, Rashba effect, or orbital Hall effect, many other, whatever they mean, is all is very interesting. Now, another interesting physics in TMD material is we have this very strong Coulomb interaction. For this bulk, of course, they are all charge screens. So, for example, this exciton, usually room temperature they cannot observe. But in all this one monolayer case, for example, the Coulomb interaction is very, very large, so that, for example, exciton binding is very large, so that we can observe always this excitons at the room temperature. This is very interesting phenomena. So, for given this strong spinor coupling and also strong Coulomb interaction, what can we do with this diluted magnetic semiconductors? So I think this is actually, this concept diluted magnetic semiconductor is well known. People introduced the concept DMS using this 3-5 semiconductors in case of this gallium arsenide by simply doping this manganese doped. And in such a case, they can manifest this magnetic, ferromagnetic order in semiconductor. This is very important because this can be used for this spintronics and with a very ultra low power consumption. This is one of the key concerns. But unfortunately, this Curie temperature is very low. We can operate at room temperature. And the gate tunable, of course, because it's a semiconductor, they can do that. So they can engineer all this switching again using this only spins. It's very good. But because of the limited Curie temperature, people also explored many different semiconducting materials like all this cadmium telluride or this mercury telluride and so on. But again, using this manganese doped, still the TC even worse. So they can play with it. They cannot. And they also explored for many oxide materials like nitrides and oxides. In such a case, they can show this very high TC over room temperature. But because of this insulator nature, we cannot have this gating tunability. So recently, people talk about this, all these 2D layered materials, Van der Waals layered materials, and unfortunately, all this TC is very below this room temperature. So, and of course, all these magnetic semiconductors itself is interesting, but the material is limited. We cannot explore this high TC under any circumstances that we're talking about. So all this DMS in 2D is very interesting because of the rich material library. We can play with many different materials. The problem, of course, if you consider all this semiconducting TMD material, there are varieties we can choose. And in this case, we talk about all these changes in metals, so it's a heavy mass, so very strong spinor coupling. So different from this the conventional 3,5 gallium arsenide. Now, so what about this magnetic dopant? In this case, we can choose many different magnetic dopant, so there is a freedom. Of course, the more freedom actually is more problem to choose. So actually, in our case, we thought about this one and then decide to choose the material semiconductor as tungsten diselenide. There's a reason, but I don't want to discuss so much. But anyway, this homogeneity issue, so we choose this vanadium as a magnetic dopant. Now, for given this again strong Coulomb interaction plus this strong spinor coupling, that tungsten diselenide shows this strong spinor coupling. And our ultimate goal is actually when you increase this the magnetic dopant, they shouldn't be aggregated so that we cannot, in such a case, we cannot do this gate tunability. So they must be well distributed. And of course, the room temperature TC, we can operate all these spin joints at room temperature. And for all this gate tunability case, this is also long-range magnetic order is the key. So that we can have this gate modulation. These are the key issues that we can talk about. Now, whether we can make this material, of course, simply this CVD, chemical vapor deposition, is very simple. We can simply use liquid precursors, liquid soluble, all these precursors, including tungsten and vanadium, and we can simply mix them and then spin coat on the substrate, and then supply this selenium gas, and then we can simply make this monolayer film of all these TMD materials. Now, in this case, we can use increase this doping concentration, even up to 10%. They are well all monolayer, which is perfectly okay for us. But if the doping concentration over 20%, of course, there are some dendritic, even multi-layers emerges. So we limit ourselves into this system only less than 10% of vanadium. Now, of course, we have to identify what kind of the vanadium absorbed in the system, and from the TM images, we can identify the difference of the intensity period. TM intensity from tungsten and vanadium. We can distinguish clearly. This is certainly the case of this presence of vanadium as a substitution of tungsten. So it's good. And you can actually see all this red color shows this vanadium distribution. You can see clearly they are well uniformly distributed in the system. And this is the case of 2%. But if you go further with a different doping concentration, still vanadiums are well distributed in the whole system up to very high percent of 10%. So this is good. And of course, we like to show this dynamic semiconductor. In such a case, we have this P-type with vanadium doping. As you increase the vanadium doping, of course, this on current increases because increased carrier concentration, and if you increase the vanadium doping concentration much higher, like 10% or 20%, then the semiconductor become highly degenerate, and this behaves like a metallic behavior. So we try to limit the doping concentration below 5%. Now, we first like to demonstrate this magnetism in the system in using magnetic force microscope at 0.1% of vanadium doping. And you can see this clearly magnetic domain stripes, which is very particular, but usually it can happen also in many other systems as well. And if you change this temperature and you clearly see these magnetic phases shift, that means all these are really this magnetic system. And you can see this the MFM phase. We clearly see different phases at the different positions, and as you increase the temperature, they become sort of saturated, and this phase no longer shows this magnetic behavior. So I think it's still 300K is perfectly reasonable to show this magnetic order in the system. So now what about the gating? Of course, we can do gating. In this case, simply by using this MFM, we have this backgate, and we can simply have this sort of this gold tip on the particular position. We can do gating, and you can see this all these even at the room temperature we have this magnetic domain changes. So which is the key for this gate modulation. Of course, at a high temperature, I mean it's at minus 10 volt, we have different temperature. Of course, then that's again this magnetic domain disappear at a low temperature. I mean, this at a different temperature. So this is good. But still, people worry, okay, you are only talking about on a very small area by this FM tip. People usually wonder, if you do the real device, would it work? So this is the question. So we actually try to demonstrate this the whole material using this circular polarized light. In this case, we usually cover above like a few micrometer sample. And in such case, for this particular this tungsten diselenide has this K and K prime. We have a spin up and spin down because of this valitronics. And we can particularly assign this spin systems with impurities, with vanadium, with different spins, we see different band structure. In such a case, if you measure this circularly polarized light for this excited either sigma minus or sigma plus, we can detect this sigma minus and sigma plus and see the difference between these two. And you see this polarization, particularly emphasizing this excitons. We can see significantly the signature of this magnetic domain in the system. This is all done at the room temperature, so at least we can demonstrate this magnetic order persists at the room temperature in this case. Now, whether this one, all this vanadium doped tungsten diselenide system has this, whether this long-range magnetic order or not, we can simply plot this density profile, spin density using this DFT. And you can see around this vanadium, we have very strong ferromagnetism. What's more important, if you go further away from this vanadium, you see all these tungsten sites spin ordered, very long, very far away from this vanadium. So we understand this in terms of this rather strong, this, I mean, this D in the tungsten and D orbitals in also vanadium, so which is very different from this conventional gallium nitride, gallium arsenide system. And we believe this is because of strong SOC, also this strong Coulomb interaction that persist, the magnetic order persist very far away from this vanadium. So this is actually the key to realize all these spin joints at room temperature. So for our system, all these questions, I think, have been well resolved. So this is very nice. And then what else can we think about? Of course, vanadium, when you use vanadium, there is a weakness. Magnetic moment is very low, only one Bohr magneton, which is very smaller than other, like gadolinium and another magnetic system. So whether we can add up some additional magnetic order in the system, in this case, we talk about this selenium vacancy in the system, because selenium vacancies are very rich everywhere during this CB deposition. So whether this additional selenium vacancy provides any magnetic order, in such a case, usually we can observe this selenium vacancy from this IV transport, but by simply using this optically activated system, by just light illumination, we can activate this selenium vacancy, and by fixing this voltage, we can do this magnetoresistance measurement, and you see this hysteresis clearly shows near this this selenium vacancy states, whereas the rest we don't see them from others. And you can see this spin density at different gating. You see around this ferromagnetic order expands further with this gating, which is similar to this case. So apparently, the selenium vacancy does provide this magnetic order, which is also very interesting because they can play with it. Now the question whether selenium vacancy involved with this vanadium together then show any further synergistic effect. So we measure this again, this magnetic resistance. In this case, this is this S treated sample, the CVD sample. We show this almost no magnetic hysteresis, magnetic resistance hysteresis. But if we anneal the sample, like 500K at in the vacuum chamber, and you see, we imagine this selenium vacancy vaporizes so that we can see this magnetic resistance is very clearly shown due to this increased this magnetic order due to this presence of this selenium vacancy. And as you see this temperature dependence, all this butterfly ML hysteresis shown, and it's quite persistent up to room temperature. So this again shows this importance of this additional selenium vacancies that involves this magnetism in the system. Now we can also see this MFM, so see with a temperature different annealing, and also this measurement temperature, and all this brightness actually is related to this magnetic phase, and you see this contrast quite persistent up to this room temperature at high annealed temperature. Of course, at the very high annealing, of course, sample become deteriorated. We do lose all this information from the magnetic system, and you can see this magnetic phase. You can see this all this positive, which is related to this this magnetic order, and you see as you increase this annealing up to 500 Celsius, and you can see that they are quite persistent. Whereas at the very high annealing temperature, of course, sample become completely destroyed. Now, we can actually look at this TM, see what's going on, and there are many different cases. As you increase the temperature, of course, all these different impurities related. We have this all perfect vanadium selenide, and also we have single selenium vacancy. Each vanadium has 6 selenium. I mean, 6 vanadium has 6 top and bottom atoms, selenium atoms, and in this case we have 1 selenium vacancy. But as you increase the annealing temperature, not only this guy, but also we have like 2 selenium vacancies around this vanadium, and even 3 selenium vacancies around this vanadium. So this is certainly shows this high magnetic order as you increase this annealing temperature. So from this analysis, we can see, of course, we have this vanadium. Around this vanadium, we have the selenium vacancy case. And just adding this selenium vacancy around this vanadium shows this additional this magnetic moment in the system. So whether we can, of course, this is all sort of a hypothetical. So we introduce this DFT calculations, and up to pure, of course, material does not show this, I mean, the high magnetic moment because of this 1 vanadium. We have this 1 Bohr magneton, and we increase the selenium around. That doesn't work. So still this 1 vanadium. And then if you introduce additional, I mean, this, we even for this one, I'm sorry, this is a pure tungsten system with a selenium vacancy. It doesn't work, no magnetic moment. But if you, I'm sorry, this is, yeah, okay. So, okay, then when you introduce this vanadium in the system, just 1 Bohr magneton. But if you increase this selenium vacancies, like 2 vacancies around this vanadium, it certainly elevates this magnetic order 1, 2, 3, and then if you add another 3 selenium vacancies around this vanadium, this magnetic moment increases up to 5. So this is quite interesting. All these magnetic moment been escalated from 1 to 3 to 5. And how does that happen? Actually, this is understood by this chemical potential engineering. So when you introduce this vanadium, the selenium vacancy in the system, the Fermi level is shifted, and then we have this spin down system goes up and become unoccupied, and then this one is shifted, flipped to this spin up, and then become parallel. So we start increasing this spin system by flipping, and by increasing this selenium vacancies more. So this is simply this chemical potential engineering by this selenium engineering. So whether this can be persistent very long, we don't know. So we explored from theory whether this high magnetic order is maintained at a very high distance from vanadium. And we found this sort of a critical distance up to like third nearest neighbor from vanadium, which is very important, is very long. So this is again because of this spin coupling, strong spin coupling, also this Coulomb interaction in the system. So you can see by introducing this more selenium vacancies, all this ferromagnetic order increases in a far distance. So this is again this reason why we have this increased TC in the selenium vacancy system. So this take-home lesson, of course, by cooperating this selenium into the system on this vanadium system increases magnetic order in our material. So so this is another sort of how long do I have?
Maybe 2 or 3 minutes.
Oh, okay. So, but I think it's enough. We can explain. So there is a special case where this the doping concentration is very small. So in such a case, we shows very unusual phenomena. If you have this resistance with a temperature, usually at a high temperature, of course, this is shows simply semiconducting behavior. But as you decrease the temperature further, then something strange happens, like large magnetic fluctuation. So this is we cannot explain this, how that happens at a very low temperature. Then we show the sort of this empty curve, which like still multi-domain this ferromagnetic order in the system. So if you take the derivative, we see this fluctuation is previous everywhere around this particular region, and we see this simply this resistance measurement as a function of time, and you see this sort of noises in the system, but this is well known, this random telegraph noise, and you can see clearly these two different states, magnetic states. Okay, so this is very interesting. So of course I didn't realize what to do with this random telegraph noise. So we got this collaborator working on this one, and so for this, usually this low frequency noise analysis can be done using this what they call this generation recombination noise or random telegraph noise, because in the semiconductor case, they have this trapping and detrapping of the defects. In such a case, we can have this telegraph signals on and off and on and off like this. And in such a case, this can be featured by simply 1 over f square in the random telegraph noise. But there is another where case is flicker noise that shows sort of a linear 1 over f. In such a case, just a multiple of all this 1 over f square because of the multiple traps. So Again, this Warner Web square simply shows this single trapping semiconductors. Now, this is different from in our magnetic system, where it still shows all these Warner Web square, things like that. But again, this is all related spin parallel and anti-parallel system. So we can see that clearly, depending on this temperature, we have two different states. Sometimes they look like two, but sometimes they are even more. So it then means it has many multiple spin system in our system. And by combining all these, the difference between the two states, this is then we define as delta R. In such a case, all these materials in the system, and you see very small delta R. Usually, this random telegraph noise can be also shown in a defect system, but usually this delta R is below like 1%. In our case, still this in our magnetic system, even though this is defect related, but still this delta R is kind of still large. But of course, if you go this system in the middle of the temperature, then this delta R become very significant, which is never been shown in the semiconductor system. So in this case, we explained that in terms of this, at high temperature, we have this intralayer coupling. So they are all these magnetic domains interact with each other with the defects. And as you decrease the temperature, of course, then this random telegraph noise in the intralayer, they become weak, and this interlayer intercoupling becomes strong. Eventually, we have this very strong delta R by increasing this magnetic domain in the system. So this is very well-known. I mean, this is actually new to interpret all these magnetic order fluctuations in the system. And what is good is actually we can have sort of switching in the network by simply injecting these electrons into the system, and they have this spin transfer torque, and then giving different magnetic order in the system. This is a p-top, so if you go this opposite, of course, then they are different because it's electron injection into this p-top system is different. So we have very switching of this resistance depending on this positive bias and negative bias. We see clearly this polarity changes. This can be used as a sort of switching using this random telegraph noise system. So I think this is very interesting. But again, it's interesting, but if you use different doping concentration like 0.5%, then it's very amazing to have a very unconventional this MR hysteresis. Usually at a low temperature, we don't see magnetic hysteresis. As you increase the temperature, all of a sudden, this magnetic resistance hysteresis is flipped from one to another, and even higher temperatures quite persist even at the room temperature, and even above this room temperature shows this very high magnetic resistance hysteresis. So this is actually very unconventional, and we still don't know. And if you look at this particular case, this is quite linear, which is again never observed before. We believe this is related to magnetic topological insulator, but we have to work on this more. Well, it's certainly very interesting. So anyway, I'd like to finish up my talk. Whatever we start with is diluted magnetic semiconductor using vanadium doped tungsten diselenide. We can demonstrate all these TC temperature above room temperature, and we have gate tunability also is possible. and sample vanadium segregation is completely removed. So all these now give some room for this spin strength in the future at the room temperature. So certainly in all these TMD materials, we still talk about this strong SOC and Coulomb interaction, and that's actually very important for many interesting physics phenomena. So I think I'll have to stop and questions, okay?
Thank you very much, Professor Lee. One probably quick question. Maybe I can ask one then. So you show that as a function of temperature, the local coordinations are changing. You have different sort of vanadium coordination environments. Is there any trend, like for example, if you increase the temperature for treatment, do you have a higher degree of sort of vacancies?
The number of vanadium atoms are fixed, but cell vacancy.
Yes, that's right.
That's easily, and this is what happened, and that's what we observe from TM. So for fixed this vanadium, of course, around this vanadium, cell vacancy vaporizes, and then that induces this additional magnetic order in the system. This is what happens.
Okay. If there are no other questions, then let's thank speaker one more time. Thank you very much, Professor Lee. So I will then hand the stage over to Jamin to close the symposium.
I'm just chairing the closing session,
chair the
meaning that my role is just passing the mic to Xiaodong. It's my pleasure to introduce Professor Xiaodong Chen for the closing remark.
Very good afternoon, everyone. I mean, it's my great honor to be here to do this closing remark. Okay, so yeah, it's really come to the end of this summit. So I really want to express my great kind of appreciation to everyone involved, make this event a success. For all our distinguished speakers, lead research, demonstration, incredible capability or potential of the nanoscale and material chemistry to address the global challenges such as sustainable development and also healthcare. And also for our organizers, including ACS publication like Amy, Terry, and also our local organizers, Jiaming, Sonju, and also the section chairs, also our KCS local staff. They spend a lot of time already to plan this event. So this is something also very, very important. Let me share one example for the live webinar. In fact, there are not only about the participants, not only about in this room. In fact, there are more than 10,000 audience outside of this room to really live watch this summit. So this is kind of, I mean, before pandemic, right? You never have this kind of hybrid motion, this kind of mode, right? But now it's happening nowadays. Okay, so this is also because these kind of organizers really spend a lot of time. You can see involve so many people here. But of course, also very important to make this event success. Also, our S-team participants still stay from beginning to now to the end, to participate in this event. This is also very, very important. In fact, the post-pandemic world really present various challenges and also exciting opportunities to really for all this kind of research community. I mean, for me, I feel very, very important is collaboration. This is something really can help to address all these kind of issues. But of course, this collaboration involves interperson collaboration, interdisciplinary collaboration, inter-country collaboration. Also very important, inter-society collaboration, right? Like this KCS work with ACS. These are examples, right? I mean, for ACS publication, we have very big kind of mission. It's for training. It's education of our authors, our next generation authors, our next generation editors as well, to really, next, in a few years later, they can really carry on So this is something I think for ACS preparation will be play more important. And also, I think, Amy also have kind of some planning already to really how we closely work with our Korean colleagues in here. But of course, we know already, now with this kind of rapid development of a large language model, like for example, ChatGPT, right? In fact, this really imposes a big challenges for our research, for our education already, right? I mean, especially how we can really nurture our next generation students, not just, teach them not enough anymore, right? How we can enable them more with critical thinking, innovative mindset, and also most important, human touch. This is something the machine cannot enable you. Only the human can enable you. So this is something one important, very important. This also gives us a lot of challenge as well. But we are really confident as this whole community come together, we can address these all challenges. So in short, stay curious, stay connected, stay inspired. Thank you very much. Have a good night.
Thank you very much for your attendance. Then for the distinguished guest,